భవాని
హలో సార్.
అమ్మా ఈ క్లారిటీగా వస్తుందా వాయిస్ క్లారిటీగా వస్తుందమ్మా?
వస్తుంది కానీ సౌండ్ వస్తుంది సార్. 2 డివైసెస్ ఉన్నట్టున్నాయి. ఒకటి మ్యూట్ లో ఉండాలి కంప్లీట్ గా.
ఒకటి మ్యూట్ లోనే ఉంది.
ఓకే సార్.
ఇప్పుడు రికార్డ్ ఇప్పుడు ఎందుకమ్మా? రికార్డింగ్ స్టార్ట్ అయిందో పెట్టుకుందాం.
ఓకే సార్. లైవ్ లింక్ పంపించండి సార్. యూట్యూబ్ లైవ్ లింక్.
ఇండియా మ్యాప్ ఒకసారి
ఇప్పుడు వాట్సాప్ పంపామమ్మా భవాని?
హలో సార్, చెప్పండి సార్.
వాట్సాప్ పంపావు కదా ఇప్పుడు? ఓకే, పంపించేస్తున్నా. సువర్ణ, నువ్వు ప్లీజ్ ఆపురా. నీకు జాయిన్ కూర్చొని భవాని ఒక్కసారి వినాయక కుమార్ సార్ జాయిన్ గా ఉన్నాను.
ఓకే సార్.
Sir, namaste, sir.
Namaste. Good morning, Karnakar. How are you? Good morning, Anuradha ma'am. And
Morning, sir.
Hello. Yeah, I'm audible, right?
Sir.
I'm audible, right?
Yeah, sir. You are audible.
Thank you very much.
Good morning, madam. Hello. Bhavani madam. Sir,
Yeah,
when
Good
hello,
morning,
good
sir.
morning,
Good
sir.
morning.
వినిపిస్తుందా?
Yes.
Sir, good morning,
Wait
sir.
for
Okay.
1-2 minutes, sir.
ఓకే అదే నేను 10 అంటే అప్పుడు అందుకే గుడ్ మార్నింగ్ అని చెప్పేసి.
Good morning, Vinay, sir.
మేడం గుడ్ మార్నింగ్ అందరికి. గుడ్ మార్నింగ్ ఆల్ ద principals, sir, organizers and the participants and the delegates from other.
Already guest Dr. Praveen sir joined, sir.
Yes, I saw. Yes.
జాయిన్ అయ్యా. హలో
Sir, good morning, sir.
Good morning, sir.
Hi, sir. Good morning.
Sir, shall we start? Principal, sir.
Hi, Praveen ji. Very good morning, sir.
Namaste, sir.
Okay, sir. Start.
Okay, sir. Good morning, everyone. I am Vinayak Kumar from Government Degree College, Sadasipet. We are very much pleased to have your presence on virtual mode from the nook and corner parts of our Telangana, and we are very much pleased to have in our presence Dr. M. Praveen sir and Dr. D.T. Chari sir from Commissionerate of Collegiate Education. I take this opportunity to give in brief about the organizers of this webinar, that is National Workshop, which is funded by RUSA. And this is in Dr. D. Karunakar sir's career, and this is, I think, 4th or 5th national workshop. And sir conducted 2 national seminars at Tara College and 1 at Government Degree College, Sadasipet, in 2019. And this webinar is being organized in collaboration with our botany department, that is our madam, Srimati Anuradha madam. And the topic of this workshop is advanced technologies in biological and chemical science. And for this inaugural session, I am very much pleased to invite, actually we are expected to have as chief guest Sangareddy District Collectorate sir, Sri M. Hanumantarao sir, but for their busy schedule, we are unable to make him join. And we have guest of honor, Dr. M. Praveen sir, Joint Secretary, Telangana Social Welfare Residential Educational Society Institutions. And special guests are from our Commissionerate, Dr. D.T. Chari sir, as Academic Guidance Officer for us. And in this context, I request our principal sir to speak few words. Over to Patanjali sir, principal sir.
Namaste. Greetings to all who attended today's seminar on advanced technologies in biological and chemical sciences, organized by the Department of Chemistry through IQAC with our college botany department collaboration and funded by RUSA. First of all, I would like to express my heartfelt thanks to Sri Naveen Mittal Garu IAS, the Honorable Commissioner of Collegiate Education and the Director of RUSA. Sir made many progressive, significant changes in our collegiate education. Commissioner sir approved our seminar along with the funds as soon as we put in the application. This seminar is not without SAR's permission. So many heartfelt thanks to the Commissioner Sir of Collegiate Education as college principal. I extend my thanks to Chief Guest Sri M. Hanumantrao Garu. When we appealed together in the matter of our college drainage, it was immediately approved by the Honorable Collector and funded. The work was also completed. Therefore, I would like to express my heartfelt thanks to him as principal. I extend my thanks to special guest Dr. D.T. Chari, AGO CCETS, for attending this seminar as soon as we requested, by responding immediately to any inquiries we may make by phone or message. Thanks to him. I extend my thanks to Guest of Honor, Dr. M. Praveen, Joint Secretary, Telangana Social Welfare Residential Educational Society, for attending this seminar. And the Chemistry Department of the college have organized about 10 such international, national, state level conferences in our college so far. On this occasion, I would like to thank once again to those department in charges, Dr. S. Devi Ramani, Dr. D. Karnakar, and the faculty and non-faculty staff for providing the appropriate encouragement and assistance in the conduct of the seminar. I would like to extend my best wishes to the keynote speaker, resource persons, principals, lecturers, students participating in this seminar from all the different colleges. Once again, seek your cooperation to make this seminar a success till this evening. Jai Hind. Thank you. Hello. Hello.
Vinayak sir, you are in mute. Vinayak sir, you are in mute, sir. Yes.
Okay.
Please.
Okay. Now is it okay, sir?
Okay.
Okay. So this is what we experience in these virtual webinars. Sorry for that interruption. And it is my pleasure to introduce our CC representation, Dr. D. Thiruvengalachari sir. And we are very much pleased to have in your presence, sir, in this workshop. And I will give a very brief introduction I have with me. Sir completed his MBA, MPhil, PhD from Kakati University, and presently working as academic guidance officer for through Commissionerate of Collegiate Education for Telangana degree colleges. And sir published 35 research papers, and sir has so much of credit by publishing his papers in India and abroad and also in international journals. And sir attended 22 international conferences, and sir's credit, sir published 45 articles and guided 3 to 4 PhDs. And sir has published, conducted 3 national level seminars. And sir, Chari sir completed UGC project in 2013, funded by UGC New Delhi. And sir got Yuvatarangam project, best project from Telangana College, government degree colleges in 2015 and 2017. And sir also conferred with the Best Teacher Award in 2017 and 2021. Chari sir, we are very happy to have your presence for this webinar, and we are very much pleased to have to listen few words, as you are the one of the academic guidance officer for us from Collegiate Education. And over to you, Chari sir. Thank you.
Thank you, sir. And good morning, all.
Good morning, sir.
President of the program, Dr. Patanjali Garu, Principal of Government Degree College, Sadashivpet. Program Coordinator, Dr. Karunakar sir, Assistant Professor of Chemistry. Today's guest of honor, my friend, Dr. Mamidala Praveen, Joint Secretary, Telangana Social Welfare Residential Degree Colleges, and other organizing committee members of this virtual workshop, teaching and non-teaching of staff of Government Degree College Sadashivpet, all the participant friends, very good morning one and all. I'm very happy to associate myself with this workshop being organized by Government Degree College Sadashivpet. Right from the pandemic 2019, for the last 2 years, Government Degree College, they are taking the advantage of this pandemic situation and conducting many number of programs, because it has become very easy to get the resource persons. We can get the resource person from nook and corner of the country as well as from the abroad also, without any cost and without any time efforts. Our Naveen Mittal sir always says that, our commissioner sir, comparatively physical seminars, these virtual seminars will certainly cut down the cost of organizing seminars as well as spread the message or information or the inputs of the seminars to the wide sections of the society. Because the people are participants from anywhere in the world, they can attend the seminars, they can listen to the participants as well as the resource persons' ideas. Today, I came to know that resource persons, they are attending from different parts of the country. Very happy, a small college like Government Degree College Sadashivpet is coming forward to conduct a workshop like a typical nature, like it is a collaborative program within the college itself. At the two departments, they are coming together and they are conducting a program like Department of Botany as well as Department of Chemistry. They come together and conducting the program. It's a very good sign. If all the departments come together and if they work for a noble cause of the academic excellence, certainly we can expect a better fruits from our different various sections of the society. Similarly, the topic of today webinar is very apt to the current pandemic situation. Advanced technologies in the area of biological as well as chemical sciences. Probably, as far as my knowledge, being a commerce faculty, I don't know much about, because Mohammed sir should say something about this, the crux of the and importance of the very topic of the seminar. But as far as my knowledge is concerned, both the areas are quite relevant to show some solution to the burning problem of COVID, biology as well as chemistry. Even they can avoid some fear which is there at least among the people in the society. COVID pandemic. So this type of seminars and deliberations in this platform certainly will pave a way to solve certain problems of the society also. Really, I compliment the organizers, the principal and the entire team of Government Degree College Sadashivpet for coming forward to conduct such webinars. So I congratulate once again an entire team for coming up of these webinars. Hope so other colleges also should get inspired with this program and hope that this will become a success and real deliberations will take place throughout the day. Thank you for giving me this opportunity. Thank you all.
Thank you, Chari sir. Once again, I extend my regards and wishes and greetings on behalf of our principal sir, teaching and non-teaching staff, and also the organizers of this national workshop. Thank you very much, sir, for being with us in this national workshop. Thank you very much, Chari sir. And now I invite to this webinar our guest of honor, Dr. Praveen Mamidala sir, currently working as Joint Secretary, Higher Education, Telangana Social Welfare Residential Educational Institutions, Government of Telangana. Sir worked as Associate Professor in the Department of Biotechnology, Telangana University, Nizamabad, and also postgraduated from sir taught at deemed universities like Postgraduate College of Science, Saifabad, which is affiliated to Osmania University. Praveen sir achieved distinction with university rank both in MSc Botany, Kakti University, MTech Biotechnology from JNTU Hyderabad, and Sir completed postgraduate diploma in patents law from prestigious law university, that is Nalsar, Hyderabad. Sir received several national and international fellowships, international training course at Biological Research Center, Szeged, Hungary, under ITC fellowship. The World Academy of Sciences, Italy, Chinese Academy of Sciences, CAS, China Postgraduate Fellowship in 2004, and TWACAS Postdoctoral Fellowship in 2008, UGC Postdoctoral Fellowship in 2006 and 2007. And much to his credit, Dr. D.S. Kotari Postdoctoral Fellowship in 2009, and Ohio Agricultural Research Development Postdoctoral Fellowship tenable at the Ohio State University for a period of 3 years. And I think these are the very achievements that I have with me. And Sir developed so many research talents and experiments in his career. During his doctoral and postdoctoral work in China, that is from 2005 to 2006 and 2007 to 2008, Sir developed transgenic tomatoes that showed resistance to high salt concentrations. And during his second postdoctoral work, that is in March 2009 and 2010 at University of Hyderabad, under guidance of Professor Appa Rao Podile, Sir developed nanoparticles for post-harvest management in tomato. Sir also developed huge genomic resources for ash species, emerald ash borer, bedbug, potato leaf hopper, red palm weevil, using high throughput sequencing methods, that is 454 Firo sequencing and Illumina platform, during his third postdoctoral research at United States of America. Till date, Sir has published 47 research articles in his high impact factor journals with total citations of 866 with H index 18. In the recent past, Sir received grants from Department of Science and Technology, New Delhi, antennal transcriptome of red palm weevil, worth 48 lakhs, and several other grants for organizing seminars and conferences, Indian Academy of Sciences, Indian National Science Academy, Telangana Academy of Sciences, Telangana State Council for Higher Education. Besides the major projects, Sir laboratory is funded with SERB Young Scientist Postdoctoral Fellowships and DST Women Scientist Fellowships. And Sir has presented several limited talks, 52 around in national and international conferences, symposia. And Praveen Sir is the reviewer of several peer-reviewed scientific journals, PLOS1, Insect Molecular Biology, Journal of Insect Physiology, Plant Cell Tissue and Organ Culture, Plant Molecular Biology Reporter, etc. And Sir served in various administrative positions at Telangana University, Nizamabad, such as Controller of Examinations, Coordinator UGC and Non-UGC, and Director for SC/ST Cell, Member Secretary for IBSC, Member Secretary for Institutional Ethical Committee. Sir, Praveen Sir, as the Joint Secretary from Telangana Social Welfare Residential Educational Institutions, and just now what Chari Sir said, and this is our very remote and college with the confined strength, and we are very much pleased to have your presence, Dr. Praveen Sir, and for this webinar, that is Advanced Technologies in Biological Chemical Science. Once again, I welcome you to this webinar, sir, on behalf of our principal, organizers, teaching and non-teaching staff, and all the participants in the webinar. Over to Dr. Praveen, sir.
Thank you very much, Kumarji. Good morning, one and all, my dear friends. I'm so happy to join this seminar with my old friends, Dr. Karnaka, who is my batchmate while I'm doing my master's, and also with my friend, Dr. D.T. Chari, who is my colleague, my initial stages of my teaching. So I'm really happy and glad to see senior most scientists delivering their valuable messages like Dr. K. Sureshji, Professor G. Rambabu Garu, Dr. D. Ramarao. Again, this Muralidhar, my old friend, he came from Kakatiya University, and Prashanti, again, she's a batchmate of mine. So I can see all my old buddies in this meeting. And also I can see very good teachers and students in this group. First of all, let me thank the chairman of the seminar, Dr. Patanjali Garu, the principal of Government Degree College, Sadasiva Bhet, and Dr. D. Karnakar, the IQAC coordinator, who invited me for this meeting, particularly on this national seminar on advanced technologies in biology and chemical sciences. And also I thank the teachers from botany department who tried to combine the both chemistry and biology, which is really needed. And friends, before I say something, I should say I'm a teacher, basically. Of course, now I'm doing administration. I'm a teacher, a biology teacher, again, with a very little knowledge in chemistry, and turned into administrator in the recent past, but still loves teaching and again doing research, because whenever I get a chance, I jump into my colleges and I start teaching, whether they're here or not. I go on teaching for hours together, because it's my passion of, it's just like a passion, that's all. So after a long gap, I got an opportunity to share a few of my experiences, which are really in line with this ongoing seminar, because it's something very important, I can say. I will take not more than 10 minutes, my dear friends. I will stick to my time. But the real importance of the seminar, if you go or if you look into any Nobel Prizes awards in the recent past, or say since last 3 or 4 decades, whatever, particularly in the area of medicine or physiology and chemistry, majority of Nobel Prizes, they go for chemists who look into biology, or for a biologist who are trying to solve their research question with the chemistry. So it's kind of a combinatorial approach, where a biologist use chemistry or a chemical guy use biology, and such people only will get Nobel Prizes. If you see the recent past, very recent Nobel Prize discovery in medicine goes to David, Julius, and Adam. So their discovery was on receptors for temperature and touch. So it combines physics, it combines chemistry, and again combines biology. So there's very classical experiment. If any students are really attending this, I suggest them to visit now and then the website of Nobel Prizes list. So where you can find how they discovered some innovative things. If you look into this recent discovery, how they used the capsaicin from chili peppers to identify an ion channel. So how it gets activated, and how you perceive the painful heat. So only thing is, the chemistry guy always think that biology is tough, unfortunate. And biologists think that chemistry is something Greek. Why I'm saying is several doctoral students, I can say doctoral meaning the stage of PhD, even if you go back to Usmania or Kakatiya or even University of Hyderabad. So they are very good in synthesizing a very good molecule. Say the molecule has got a potential anti-cancer or antimicrobial, or say antiviral or antifungal, whatever. But when you ask them to do antimicrobial test, they don't do. They simply come to my lab, say, brother, can you help me, friend, can you help me in testing this antimicrobial activity or not? So it's very simple. They don't want the principle behind the antimicrobial activity. Just they want to know whether the compound has potential antimicrobial or not. That's all. If it is antimicrobial, they say plus, or if it is not, they say minus, and they record it and they publish the data. Then they want to say that this is antimicrobial. This is a compound which contains huge antimicrobial compound. But they never look into biology, like how it really works. For example, if you say penicillin, I can see whenever I attended some chemistry classes, not attended though, but when I attended some seminars, it's a huge derivative, it's a huge structure. A chemist will always try to understand the structure, but he never understands what is the cell wall of bacteria, or how this compound is really breaking down, or how it is really making an antimicrobial or antifungal. That is the reason why since biologists are not looking into chemistry, or chemistry is not looking to biologist. Till date, I have several students, my PhD students. If I ask them to prepare a buffer, they are gone. They don't know what buffer is. After completion of BSc, MSc, even completion of PhD, if I ask them buffer, they are gone. Sir, buffer is buffer. I don't know, sir. I need to ask a chemist guy. I need to ask someone who can tell me what exactly buffer is. Now, what I will do is when anyone comes into my lab, I'll tell you very basic experiences, my dear friends. I teach MSc biotechnology students. So I ask them, what do you mean by 5' to 3' DNA? I hope everyone knows about DNA, right? Deoxyribonucleic acid. It's a very common word, okay? RNA, ribonucleic acid. So it comes from intermediate, if I'm not wrong. People try to study this particular DNA from intermediate, 10+2. And they also study the deeper way in the degree, and also in the MSc, and also during their PhD. But if you ask, what is this 5' to 3' DNA? Why this is prime is? Why this prime? Or you simply ask them to draw the structure of DNA. Again, they say, sir, I am sorry. It is something related to chemistry. It's not my job. It's not at all my job. Simply they escape that the structure of DNA is a double helical, but they will not look into a chemical structure of the DNA. If they really know that the DNA is a nucleotide, it contains a nitrogenous base, it contains a pentose sugar molecule, it contains a phosphate background, since it contains two different atoms, then since it contains a pentose sugar, it should be named as 1', 2', 3', 4', 5'. And why is this prime is again how it is really helpful to us in understanding the basic structure of DNA. Then only he can understand what is DNA, what is replication. Replication in biology is different, replication in chemistry is different. What exactly happening chemically? What is chemistry in transcription, or what is chemistry in translation? Or if you know all the structures, then only you understand the sequencing. Today, the topic is on advanced technologies in biology. But I'm sorry to say, my dear friends, 95% of students, particularly biology students, particularly biology, they don't understand what sequencing is. Sequencing was discovered long back, my dear friends, by two great guys. One is, I hope most of you know, Max M. Gilbert and Sanger. A great chemical cleavage method. They used some chemicals and they tried to cleave the molecule, and they understood the sequence. In the same way, Sanger was so clever guy. He tried to introduce a terminator, we say chain termination method or dideoxy method. So if you do not know what is deoxy, then how you understand about dideoxy? That is what difference it is. So simply they will rote learning. It's kind of rote. They write, yeah, this is the dideoxy method, this is the chain termination method, that they write and they pass. And they never understand the sequencing. If you do not know the sequencing, and how you understand bioinformatics, and how you understand the advanced technology, so-called as 454 pyrosequencing, discovered around 15 years back. Way back in 2010, I used to play with the sequencer. But now again, till date in India, people are they don't want. Okay, मेरा पास sample है. I'll give the sample. Please sequence it and give the sequence. What is that? What is that you would really understand if you just get a sequence, if you do not know the basics or fundamentals? So this is what we really miss. We really miss. Since we have some biology teachers here, I think Anuradha is a botany teacher. If you ask a student isolation of DNA, they don't do. They do not do it. To isolate DNA, now we have very senior scientists here. They are very familiar. We need say 3 steps, or we need say It's a three-step procedure. Say we are going to break down the cell wall, we are going to extract, and we are going to purify. These are the three steps. Where in the third step, use a buffer. As till date, I ask my MSc students, friends, how to prepare a buffer for this, why we are preparing this. They don't know. They only know that add solution 1, wait for some time, add solution 2, make it upside and down, and add solution 3, then add ethanol and precipitate. And if you ask them why to add ethanol, what makes ethanol to precipitate, or why, they are gone, my dear friends. Please ask the senior most scientists from CSL labs to a ordinary lab in the Osmania University or say Kakatiya University or whatever. So they lack this. So at least with this seminar, I can say you should try to create interest in the students. See that they should have a great interdisciplinary approach. Why I'm telling all this is all my experience, my dear friends. Exactly 26 years back, when I met a Professor Vidyavati. Vidyavati was a Vice Chancellor of the Kakatiya University. So during my master's, I was in my second year in my master's in botany. So I went to her lab and I asked her, ma'am, I want to do an experiment. Then she told, Bete, what is this? You are a MSc graduate and you want to do research. Foolish, no, you can't do. To do research, you need to pass MSc. Then I told, ma'am, I have a really very good research question with me and I want to do that. And after several follow-ups, say 5 to 6 times, then she asked me, TK, what do you want to do? Then I told that during those days, there's a huge number of suicide attempts by the farmers. They used to drink pesticides, just like a Coca-Cola. How we drink Coke during 1990s and 1995, the pesticide farmers, particularly who are growing cotton, they attempted lots of suicides. So I thought of testing those compounds on the algae in the lab, exactly how this compound shows lethal effect on the algae. So then she told, okay, TK, jalo jao. Then I want to prepare the compound for my experiment in the ppm, in the parts per million. Believe me or not, my dear friends, we have 2 blocks in my Kakatiya University. One on the left side is the botany and the right side is the zoology. I met almost 10 professors how to prepare a ppm, a solution of like a ppm. No one knows it. No one knows it. They say that it's not my job. Oh, chemistry ka job hai. Entire university I gone. Those days there is no Google. Just now we have a Google, we have a smartphone. Whatever you want, you can type and you get the data. So I went to a professor. I still remember his name is MSK Prasad, a very good biochemist. He handled me, hey Praveen, good. At least in MSc you are trying to come up with a research question. Then he took me to his lab and he taught me what is ppm. So there I thought, yes, if you want to become a scientist, particularly say in the research field, if you want to go, you need to have a math, you need to have a chemistry, you need to have almost all the domains into your research. So that is the reason why, my dear friends, the moment when Dr. Karnakar asked me, Praveen, I want to invite you and to see that you want to share your experiences with these students and teachers. I immediately said yes. Do you know why? I thought this is a one-to-one seminar, offline seminar. And again, after some time, he called me 2 days back. He told me that, Praveen, it is a virtual seminar. So, of course, TK, as our friend Chari was telling, with the virtual seminar also, we are trying to meet from nook and corner. But still, I thought, if at all I get some time, I would like to come to colleges like GDC Sadashivapet or colleges in the remote places to tell them really how you really enjoy if you combine 1 or 2 more domains. We are going more narrow in that we are the masters in the subject, but it's not true, my dear friends. It's not at all true. If you go to abroad, if you go to western side, look into the western side, at least go to Israel if at all, if you get some time. All labs will come together and they brainstorm and they will try to innovate something new. Small coronavirus, my dear friends. Everyone knows coronavirus. No need to say anything about coronavirus. Go back to a rural space where there is no technology, where there is no newspaper, but they know at least mask. They don't know what coronavirus is doing. They don't know what's happening, but they are now fear. Without of fear, they are wearing mask. Out of fear, they are really doing the sanitation. So, but we do not have much clues on what exactly coronavirus is. As a scientist also, we are not aware like what this Omicron is doing now, variant of concern, what we say. Lots of technologies we have. The only reason, friends, is we don't understand the biology of the virus till date. That is so unfortunate. So it is still naive. It's real because if you see a virus, I say it's a chemical. If you see it, just because it contains a protein or it contains a nucleic acid, it may be a kind of RNA or DNA, and enters into the host and starts damaging the host with the host enzymes, and it will enjoy, replicates, multiplies. But how to control such a virus is again a chemist. The chemist has to look with the biology view. How can I how best I can come out with some retro or the antiviral drugs. So it's a challenge. So unfortunately, is the science is not taken up so much serious in our India, I can say. The only thing is they never develop a question right from their childhood. Because to my knowledge, way back 30 years, because now the same is happening. After 10th class, they want to take up BPC to become a doctor. Please remember, they never take BPC to become a scientist. They take a BPC to become a doctor, or they take NPC to become an engineer. If they don't get through, they join in some sciences, and they don't know why they take up chemistry, and they don't know why they take up biology or botany, zoology, microbiology, or whatever. Just like that, they'll take up, and they'll end up with a master's. Why they don't know why they are doing the master's. And they do PhD just because it's like a crown, and we have to do some PhD and get some papers published, get some job. That's all. So you never start asking a question, the research question. This is a very important. Wherever I go, I'll always ask my students or my friends or my teachers to make our student community to come out with some out-of-box ideas and try to test and try to do some experiments. Science is all the observations, my dear friends. It's all simple. If I say about science, it may be chemical sciences or biology or whatever, physical sciences, whatever. It's all observations, experimentation, a great methodology, a hypothesis, something, you do great, you can innovate, saving the society. So ultimately, what I want to say is a chemist should become a chembio, and a biologist should become a biochem. And I'm sure in coming days, we can come up with some good innovations. So before I close, I really thank the organizers, Dr. Karnakar, Dr. Anuradha, and the principal, and all the teachers behind this project. It's not that much easy, my dear friends. Though it's a virtual seminar, organizing virtual seminar, inviting the resource persons, the big resource persons from different places. I can see a very big shots like Dr. K. Sureshji, whoever, like a principal scientist in CSL labs, taking their time, inviting all the other resource persons. It's a very tough job. So it should happen. It is not, since it is as our Chariji is telling, is a virtual, it's nothing wrong having a national seminar every month. There's no need to keep a national seminar as a title, but a seminar. And as a brainstorming session, invite all the students, all the resource persons, come together and think tank and try to come up with some better research questions right from undergraduate level. This is what we are missing. We think that research is the part after MSc. No, research is right from the childhood, right from 6th class, 7th, 8th. They should start thinking. And I'm sure if these biologists and chemists come together, I'm damn sure that we will come with the better innovations in the near future. So thank you very much, my dear friends. Thanks a lot. And I wish you all good luck. And let's hope this seminar will become a successful event in by end of the day. Thank you very much.
Thank you very much, sir, for your brief and elaboration about advanced technologies in biological and chemical sciences and highlighting the concept of research, not only in faculty and also among the students. And but nowadays the students are, as you mentioned, they think after completing BPC, go for BSc and a doctor or engineer through MPC. And most of the thing they think that only for the sake of employment as soon as they want to get. But there are so much of scope and explore in research through sciences or any other subjects. And once again, uh, we are very much pleased to have in this webinar through virtual mode. And you also mentioned the concept, the using of mask. And I think it is somewhat we have to feel fortunate that maintaining a physical and social distancing, and we are making the webinar as successful. And once again, on behalf of our principal, organizers, Dr. Karnakar sir and Srimati Anuradha madam, and the students, and on behalf of all the participants, I extend my regards. Actually, Dr. Karnakar has to brief the topic line, tagline, but in hurry, I gave the introduction of Chari sir and Praveen sir. I think with this, we conclude with this inaugural session. And over to Anuradha madam to introduce Dr. K. Suresh sir, Principal Scientist. And I request all the participants stay tuned in the webinar. Thank you very much.
Thank you, sir. Good morning, everyone. I cordially invite you all to this one-day online workshop. Actually, we wanted to make offline, but due to Corona pandemic, we have to turn out it as online national workshop. And it is my great pleasure to introduce Dr. Suresh Korpol, Principal Scientist and Head of MTCC Division of CSIR Institute of Microbial Technology. Here are few words about the Sir BioSketch. Sir has pursued PhD from Osmania University, Hyderabad in microbiology. Sir did 3 years of postdoctoral training on microbial diversity exploration and exploitation at CSIR CCMB. He visited Professor William Burney Whiteman's laboratory, University of Georgia, USA for 1 year to study about the methanogens. He pursued training at DSMZ, Germany on anaerobic microorganisms. He has been associated with MTCC and involved in preservation and supply of authentic microbial cultures to diverse researchers of the country. His research is focused on microorganisms existing in complex environments as they often require to produce bacteriogens. Sir has characterized different types of antimicrobial peptides, including antibiotics and lipopeptides, using peptidogenomics approach, where the biosynthetic gene clusters were identified from the genome sequences and characterized by biochemical methods. Sir was visiting fellow at Professor Robert E.W. Hancock's laboratory, University of British Columbia, Canada in 2016. He has 80 research papers in peer-reviewed international journals and 5 book chapters, including Burgess Manual of Determinative Bacteriology. Sir has guided 8 PhD students. He honored with awards like DBT Overseas Associateship and Raman Research Fellowship Award. JSPS invited speaker, sir has JSPS invited speaker at East Asian Young Scientist Follow-up Program on Environment and Bioengineering at University of Hyogo, Himeji, Japan. This is a brief profile of our keynote speaker, Dr. Suresh Korpol, and he has chosen a talk that is on microbial diversity, unseen treasury of antimicrobial peptides. With this brief introduction, I handed over the mic to Dr. Suresh Korpol. Sir, please carry on, sir.
Thank you very much for nice introduction, right? Srimati Anuradha Korpol, right? I just wanted to tell you all, right, that this is Anuradha is my sister, right? You know, so we are of the family, right? My father and he has a brother. So we are, 10 or 11 kids, right, in the home, right? None of them actually pursued the biology career, right? I was the first person, right? I have, we were, 3 elders to me, right? And, I was the first person, right, to pursue the biology as my career. And the only person in my entire family who followed is, Anuradha. So all the other, either they have pursued mathematics as their career, or they pursued commerce or arts as their, or some technology as career. Anyway, I thank you so much, right? I would have been very happy to come over there and, because it's my alumni, right? Because I passed by +2 from government junior college, Sadashivpet, in the year 1988, right? In 88, there was no science stream available to pursue the Bachelor of Science in Sadashivpet Government Degree College. So therefore, I went to Tara Government Degree College and I pursued my BJC over there. Afterwards, I pursued my MSc and PhD from Usmania University Department of Microbiology, and subsequently I joined Dr. Shivaji at CCMB and then moved here. At present, I am in MTCC, right? This is microbial type culture collection and national facility. I'll explain you what exactly we do in my slides. So I would recommend Dr. Karunakar and Srimati Anuradha, right, to get your students sometimes over here as a industry trip, right, so that they can actually get familiarized about the facility and the research. Anyway, I would like to share my screen, right? So I don't know how to share it. Can I say directly share screen? Okay. I'm unable to share. Sandrine, how do I share it? I mentioned, right? That. Okay. Share. Can you see the screen now?
Yes, sir.
Okay. So let's start. Yeah.
Please
So as I said, pardon me?
Okay.
Yeah, I will take probably 30 to 35 minutes, and then 5 minutes we can have for some discussions, right? And I'm always available on the phone or on email, right? If you have any doubts and if you want to discuss anything, right? So this is microbial type culture collection and gene bank. This is a facility which is actually supported by CSR and DBT, both organizations, right? So today I will explain you what exactly we do in MTCC, and at the same time what kind of research that we pursue in our lab that I'll be explaining to you people, right? So first I'll start with the microbial diversity. Microbial diversity is usually how we actually try to exploit this microbial diversity that I'll be explaining in brief, and then I will proceed with what kind of collection that we have and how we are actually trying to exploit these microorganisms, right? As clearly mentioned by Dr. Previous speaker, right? It's very clear that there are many things actually you can get from microbes, and microbes actually dictate what is life on the earth and how exactly we can proceed, right? These kind of things already Dr. Praveen mentioned, right, a while ago. But I'll be explaining in some more things in detail, right? And then we can actually have a kind of, get kind of knowledge about how we can explore and exploit these microbes for various our routine applications. So this is what is our institute in 1986, started in a small room, right, in a industrial area. And after that, 1980, we moved over here. This is our institute, right? And this is where in a small, portion we used to stay as MTCC, but now in 2016, we actually brought up our own building, right? So our division, right, in, CSR IMTech. This was inaugurated by, our Honorable ex-Prime Minister for Science and Technology, that is Dr. Harshvardhan. And he recently visited during the COVID time, right, to actually see that, how we can actually proceed, right, with certain things. And then he was actually trying to explore what kind of microbial facility that we are having in the country. At that time, actually happened to see our facility. So I would like to start with kind of, introduction that, exactly, microbes evolved on the earth, right? Before life, right? How was the earth, right? If you look at over here, the earth without life, it was almost similar to the Mars and Venus, right? So whatever the Mars and Venus composition, that the gas and other things were present, right? Similar things were existing on the earth also, right? But as earth started, life, things are different now, right? We have a lot of oxygen, nitrogen in our atmosphere, and we have many things, right, which can actually make us hostile, right, to our livable, right, sustain our life on this planet. So it started about 4 to 4.5 billion years ago. That's a long story, right? When you compare, right, average life of 80 years of an individual, right? So 4.5 billion years, this was the situation, that is Earth without life, right? But after that, right, slowly chemical evolution started, right? Many people says that what is first, protein or mRNA? Because protein, it is essentially required to, make mRNA, and mRNA is essentially required to make protein. It's like, egg or chick, right? It's again, the same problem, but many people says that it's mRNA, which actually, it started, right? Because it codes a protein, right? So it started from there. And then it actually, started after these 4, right? When we go down the line, right? It slowly started, anaerobic forms of the life existence, right? Which actually produces various gases. And some of the anaerobic forms of the lives, they actually also produced oxygen, right? So therefore, right, it slowly, the Earth started, becoming the oxygen. So from there onwards, autotrophs have evolved, and long time, right? From 4 to 2 billion years, microbes actually conquered the Earth, right? Actually, they modified the Earth into a different form. So microbes are our ancestors, right? So life started from there, right? So virus, what is a virus, right? Just now, Dr. Praveen was explaining, right? What is a virus? Virus is a virion particle, right? Without a host, right? When virus actually enters a host, right? Even a coronavirus, without infecting you, it's nothing. It's a life. It's not called as a life form. It's called as a particle. It's called as a virion particle, right? But when it enters into the host, then it starts, right? Virus has no cellular machinery to carry out its activities, but only it utilizes the host cellular machinery, right, to actually, synthesize the proteins or, are the molecules what it requires. So therefore, life started from there, right? And virus, and then bacteria, and then some archaea, and after that, slowly it started, forming the bigger life form, right? Then started the evolution. Then it has to come actually 0 years. If we say today we are in the computer era, right? Advanced life. So now, how many types of life exists on the Earth, right? As I said, that life is, Earth is filled of, various organisms, that is fungus, viruses, bacteria, archaea, right? Plants, humans, animals, right? What not? Many things are there. If you look at what is the diversity, right? Diversity is so what exactly, right? Viruses that are estimated is the 150,000 are there, right? Similarly, 100,000 or 1 million of prokaryotics exist, right? Similarly, fungus, protozoa, algae. How many organisms that we know in reality? We know very little, very, very little. We know only 15,000 prokaryotic forms, that is bacteria, right? So what is the percentage? You know, only 4.8%. But when it comes to the higher life forms, right? Higher life forms, animals, we know pretty much high, right? And plants also we know pretty much high. So we know very much about higher forms, but not the lower forms. Why? Because of the morphological features. When I say morphological features, you can identify this Jamison flower, nose flower, lion, and elephant. If I say this is not elephant, it's a donkey, you laugh on me. Is that correct? Yes. Why? Because it's an elephant. And you can go up to the form that a mold also you can recognize, right? You will say that it is a mold, but you never say this elephant is a Ramu elephant or it is a Radha elephant, right? But if I ask you who is this guy, nobody will say that is a Balachandra or Bhanumati, right? But he is very well known as, who is he? Can anyone answer? Ranbir Kapoor, right? so now you can realize that, how we actually identify, right? In the human race, when it comes to the human species, right? We are a species, right? We are actually, we are called as Homo sapien. Homo is our genus and sapien is our species, right? So we will never call him as a Homo sapien, but or we will never call him as an Indian, or we will never call him as, right, by looking at this race, right? But we only say that individually we actually identify that. But how many bacteria that he harbors? First of all, how many cells is made up of? He's made up of 1 trillion cells, 10 to the power of 13, right? But how many bacteria he harbors? Huge. It's 6 times more than actually what exactly is made up of. So that clearly says that how big is this microbiome, right? Everywhere microbes are present on the earth, right? So it's very important to understand what is this microbes, right? But now when I ask you people, right, what are these, right? These are the microbes, these are the bacteria, these are the, microbials, right? Can anyone say, when I'm asking what is this, everybody says that it's a rod-shaped bacteria. But no one can say that it's a E. coli or it's Pseudomonas or something, Brevibacterium, Bacillus. No. Why? Because it's very difficult. The morphology is very highly restricted for the microbes. Microbes have very restricted morphology. Then how to actually, determine the taxonomic status, it depends upon various techniques, which includes the biochemical, morphological, genetic features. Everything has to be compared, and then only we can actually determine what exactly is the taxonomic status of that particular organism. Right? This looks like a complicated process, but it's very easy. Why it is very easy? Because microbes have a restricted genome content. It has only single chromosome, right? It's also called a single chromosome, right? But people do not even call it a single chromosome. It's a DNA, right? It's a naked DNA present in the cytoplasm, right? At least in eukaryotes, it's separated with a nucleus membrane and called as a nucleus. But in this case, it's a in the cytoplasm. So therefore, it's difficult to identify them. But when you understand the concept that how exactly to identify them, it will become much simpler. What they do a lot of, microbes do a lot of things, right? They actually produce a lot of oxygen, right? Starting from the deep oceans. You know that deep oceans, there is a lot of life form exists over there, right? How are they getting, right, the oxygen, right? This is how microbes actually produce a lot of oxygen in the deep oceans. They are the decomposers, right? If they could not decompose the dead bodies, right, till today, you would have actually filled with the dinosaur dead bodies, right? So they are the bioremediation, right? They can actually transform all the complex materials in simple form, and they actually, convert or detoxify the chemical toxic substances in the environment, and they actually improve the land fertility, and they also involved in the crop protection. Not only that, they actually avoid the growth of others. There are some insecticides, there are some pesticides. These bacteria, when it enters into the insect, actually they kill that particular insects, or they vanish the population of the pests, right? So, and apart from that, they maintain the good health of the humans, right? There are some probiotic organisms are there. When essentially you have to take curd. A good example is, daily we eat curd. We eat so many fermentation foods. What are they? Right? There are a lot of bacteria which actually having a very good properties, right, to maintain the good life or healthy life. So that's where we are actually taking them. Some microbes, they produce medicines called as antibiotics. The first discovered antibiotic is penicillin during the World War II, which actually saved huge, millions of the soldiers' life. Right? This is discovered by Alexander Fleming. Accidentally discovered. He actually found that one fungus inhibiting his bacterial colonies. So then he found that something is happening over here, and when actually purified the metabolite, then he discovered that there's a chemical compound which is called as a penicillin, or which is named as a penicillin because it is produced by the penicillium notatum, right? Or penicillium chrysogenum, right? So therefore, he called it as a penicillin, and therefore, actually, it worked as a wonder drug. It saved huge, lives of millions of the soldiers. Of course, these bacteria actually also cause diseases. That is the most important point. Now we are struggling a lot in this pandemic. We are saying that COVID-19, corona, this. What is this corona? Corona is a virus. It's a virus. It's RNA virus, right? Usually genetic material is DNA in every life form. But viruses are exception, right? They have RNA also as genetic material, right? And when in fact, RNA will become actually double layered or double stranded, and then actually it resynthesizes again, single stranded, RNA viruses. So mechanism, it replaces the host mechanism, and then actually, it can make very much survivable inside the host cells. Similarly, bacteria also causes a lot of diseases, right? If you are taking, right, there are so many bacteria which cause dental infections, lung infections, eye infections, diarrhea, dysentery, urinary tract infections, sepsis, septicemia, right? Blood infection, right? And there are so many problems on the face or skin, this which is called as by Propionibacterium acne, right? So sore throat, etc. Right? All these problems are there. So when bacteria is actually, our microbes are playing so much of important role, then it is very important, right, to cultivate them, to grow them, to preserve them, so that we can study about these organisms, and then actually, we can understand how they are actually working as a nasty strains or some of the good strains, what is making them nasty and what is making them good, and how actually we can suppress the nasty characters or nasty features of these microbes so that we can make them as a good or vaccine candidates or etc. And that we actually study in our labs. For this, we have to grow these bacteria, right? We grow the bacteria in our labs. That's called as in vitro cultivation. In vitro cultivation is artificial cultivation, which requires a lot of media preparation, right? Every bacteria do not grow in all media. Right? They have some specific requirements. So we actually, accordingly, we prepare the medium plates and then we streak them. You can see that one colony. These all are the individual colonies. You can see clearly. Each colony, having a different features. Now you can say it's very easy, sir, to actually separate these organisms. Of course, sir, but it's very difficult because this may be different organism. This may be looking like the same, right? And this is yellow and this is yellow. They may be different, right? And when you take out one single colony, and this single colony contains about 1 million cells, you can see that, what is their size? One single colony, it's actually representative of this minute colony is also 1 million cells, right? When 1 million cells join together, we can see them as a colony. And now just you pick up this and then streak it on the other plate. When you are streaking them, right, it's a pure culture now, right? Because it's coming from a single clone, one colony. So now this pure culture, now we actually preserve them for further studies. Its preservation is very important. That is where we are MTCC, microbial type culture collection, right? What we do over here, we actually preserve these microbes in our culture collection, right? For what? For various studies, right? Different studies. How do we preserve them? We preserve them in -70 deep freezers. You can see over here, right? It's the temperature maintained within this is -70 degrees. And we preserve them in liquid nitrogen, which is actually -193 degrees centigrade Celsius, -193. We also preserve them by lyophilization, right? Means freeze drying, right? You know that seeds are, sun dried, right? What you do to dry the seeds? You actually put them in the sunlight, they will get dry because water is removed. And when you put up water, they actually regrow, they germinate. Similarly, here also, we use a freeze-dried method where we actually remove the water content from the organisms and then we made them dried. And whenever you wanted, break open, put it in the media, it will grow. So all these methods, right? But it's very important that you cannot just preserve them like that. You need a cryoprotective agent. Otherwise, what happens? The water content present actually can pierce the membrane structures. So actually, organism going to die. So to avoid this, these problems, you actually, preserve them with kind of glycerol or, other preservatives, right? Which are also called as cryopreservatives, right? DMSO is there, some skim milk powder is there, glycerol is there, etc. And liquid nitrogen is very important because these do not allow any genetic changes, right? Otherwise, here and here there is a possibility of short genetic modifications. And if you want to visit, how many bacteria we have, you can actually go to our website. This is microbial type culture collection, right? Our website is, mtccindia.res.in. We are a national facility here in India, and we are supported by CSIR and TBT. So we actually serve to the country. We actually serve various microorganisms to the industry. Why do we send it to the industry? Because industry require them to carry out some of the microbiological assays. Right? When you use a cream, let us say, if you apply some cream on your face, before that you actually have to test that cream whether it causes any mutagenicity, right? Because chemicals are known as mutagens. If it is able of mutagenicity, then it can be a carcinogen. That means cancer causing. So before that, they have to prove that. For that, a simple test is there where you use a Salmonella typhimurium, which is an indole negative mutant, right? And when you use this Salmonella typhimurium, if the substance is not actually making it to the back mutation, then actually you can use this. So for all these different assays, you need drinking water, you have a coliform test. Water should be E. coli free, right? For that, you have a standard organism with us. So you can take that and they will pursue all these things, assays. And there are so many researchers who actually depend on us for the pure microbial strains that we supply to them for the various research works. Right here you can see, right, this is the MTCC facility, right? That where we have a huge system, right, to prepare these ampoules, right? As I mentioned earlier, right? These lifeless ampoules are, made in this system, in this facility, right? Once we are done with this, we actually ship them to our, kind of warehouse, right, where we actually deposit all this or store all these ampoules, right? And these are ready to supply now to the customers. And we have various mechanized mechanized systems, right? Various instruments which are very much useful in actually identifying the bacteria or microbes. That is Biotech 2. This is actually biochemical based, right? When you put the organism, it actually undergoes some biochemical tests, right? As when it undergoes biochemical tests, then you actually, there is a color changes and which actually red with the help of a spectrophotometer at the particular wavelength, and after that it actually gives a similarity with already known species. If it is dissimilar, right, with the known one, then it will show as a new one. Similarly, a biolog. This is also based on the biochemical, right? Various sugar substrate relation. But these are not, do not identify exact identity. So therefore, what we do is, we actually use the molecular characterization, where we will take out some gene amplification. And the gene is called as 16S rRNA gene sequence, right? This is very important because this is where, this gene is present in almost all living forms except viruses, because it is essential for the construction of ribosome. That is why it is. But there is another simple way, protein-based, that is called as MALDI biotyper. What we do here is we'll actually take out the proteins and then we'll analyze, and it specifically detect the proteins which are involved in the ribosome construction. So with that matching, it actually tells what is your organism is, right? It's an easy method, right? These are the very difficult that they have actually discovered, but now it is so easy for us that just we can actually identify within a minute, right? Or at hardly 2 minutes, right? What you need is you actually keep your organism on the stainless steel plate and then you allow it to air dry. Air dry is the time what actually takes. After that, just put it in the vacuum and send the laser, it actually detects and it gives some peaks like this, and after that it will give the identity what exactly your organism is. So MDCC is an international depository authority. We preserve the microbes, right? That means we also accept the patent microbes, right? You can ask, why are you doing the research? We are doing the research just to find out what novel things that we can discover. That I will explain you in my coming slides. So in that case, if you think that your organism is having some very interesting properties, right, that can actually lead to the commercialization, right? Or you're looking at commercial exploitation, then actually you can put your organism under the patent deposit with us. So we maintain, right? How many such depositories are present in the country? There are 3. MDC is the oldest one in 2002, and there is one in Pune, which has come recently, and there is one in Uttar Pradesh, which also just in a year back is actually recognized. But only very few if you look at the countries, world, right? So India has such repositories available in the country. This gives, these are all our industry and research customers, right? So there are so many customers, right? Even Telangana also we have, plenty of customers, right? Probably we have mentioned as Andhra Pradesh, when we are not updated probably, or I'm using a Telangana, it is there, 146, right? So we have customers across the country, and they actually take organisms from us, right? So this is how we actually supply every month about 350 to 450 strains that we supply to our customers. These are the few examples of the strains, right? These are the 15 strains routinely we supply to the industry. But how many strains we have in our culture collection? We have 14,000 strains, right? 14,000 microbial strains, which can be distributed, right, to the customers, researchers, academia, colleges, right? Everywhere that we supply these strains so that they can use it for research, are carrying out some microbiological assays and various other applications. How do we contribute to the country? We contribute to the country by, they publish, by using publications, by using our strains. And they also actually, but we actually allow them to patent their organisms with us. So that is how actually we are helping the country. Right, so that is about the MDCC, right? So we are preserving these microbes. But what do we do with the microbes, right? Right. That is what the question we asked. How exactly we can use these microbes, right? Thereafter, actually, my group, right, my students, right, who are involved in these particular studies, they have started digging out how we can utilize these microbes. We can utilize these microbes for various applications, right? But what we found is, since there are antimicrobial peptides that are known very well, very much low, so we actually thought how we can use this for the exploitation of the antimicrobial peptides. Why? Because antimicrobial peptide production is a probiotic feature. So we know the probiotics. Probiotics are essential now, right? You know that probiotic means, what is that product? It is probiotic drinks are available nowadays in the market, right? There are so many probiotic drinks are coming up. So they are actually, they can use these probiotic. What they contain? They contain some organisms which produces some lactic acid and as well as some antimicrobial substances. So therefore, we thought actually, how can we improve this, antimicrobial substances production from the organisms, and how can we identify novel antimicrobial peptides? That's what we asked. For that, we actually, many probiotics, they are anaerobic in nature because they resides in the anaerobic conditions. Even in our gut also is anaerobic condition, right? So therefore, we try to isolate, some of these samples, right, in anaerobic conditions. This anaerobic, workstation. Inside there is no oxygen. So we actually, plate out all the plates, and then we identify the organisms which are actually do not allowing any other organisms to grow in their periphery. You can see the inhibition zones in surroundings. So these colonies we take out, and then we identify them, right, by 16S RNA gene sequence identity. And then we actually identify them. You can see over here, there are some Brevibacillus, there are some Lysine-Bacillus, Penibacillus, etc., right? And these organisms, I don't want to go into them too much of detail, right, because it's difficult probably to understand, right? But this is simple screening. When you plate them out, all the sample, right, some organisms they're always fighting with each other in the complex environment, like us. Right, we actually fight with each other for the place or for the food, right? Right, food definitely will fight. Here also they are fighting for the food, because you can see the growth. It's distributed growth in everywhere, right? But this organism, if wanted to grow, so it's not allowing any other organisms to grow in their periphery. This is how means when it detects something is coming over here, it starts secreting some weapon. It's a weapon, like, it's like a, machine gun, so that it can avoid all the other, colleagues, in the periphery, so that it can alone grow over here. So you can see many places like this. So it has this ammunition, right, in the form of antimicrobial peptide. So it will not allow any other organisms to grow over here. So now we actually isolate these microbes, and then there is a process. We extract these particular, substances, and then we purify them by HPLC process. And then we determine their molecular weight, and then we actually look at their amino acid sequencing by intermediate sequencing. Then we look at the genes organization, and we actually determine the MIC right over here, right, to determine their, MIC, how efficiently they can actually, work over here, right? So we also look at how they are actually inhibiting the pathogens growth, right? So we will look at how effective these organisms are, right? And how they can actually avoid the growth of various pathogenic organisms. We determine all this, right? And we look at thermal stability. It's all required essentially, right? These compounds are highly thermal stable, right? And we look at the structure. This determines the structure. The peptide is not changing any structure whenever you are exposing in 90 degrees centigrade. It's essential, right? Sometimes this kind of, thermal stability and pH stability is required because you use at various temperatures. You use in cold systems, you use in, different, hot systems, right? So therefore, it is essential for us to check, how they are exactly, the temperature is affecting them. So we also determine the structures, and then we actually look at it, how the structure is influencing the activity, how it can activate. You can see this is a protein molecule, right? The structure, when you look at it, there are some negatively charged and positively charged distribution on the surfaces. These are the crystallization process, we call it as. When you molecule, you will actually exposed to some specific conditions, it form a microcrystal, which is actually studied with the X-ray analysis, right? We send the X-ray, and then we'll determine the structure, and then we come to a conclusion that how exactly this molecule is working, right? And that when students, like one of the students is sitting with me, right? Like these people, when they publish such papers in high impact fact journals as a cover page articles, they are very happy, right? They're pretty much happy, and then they will feel that, oh, we have achieved something, right? That is what is the research. When for anything, of course, I was like that when I was a PhD student. I wanted a good paper to be published in a wonderful journal, right? So that I will, get a good position, right? Postdoctoral fellowship, and then I can visit abroad. I can go. Every dream is there. Every postdoc has, every PhD student, every research dream for this, right? That they have to, submit, go to a good conference, publish a paper, etc., right? And you can see here how the peptides are working, right? It's actually clear, just it chops off the membrane so that everything comes out. I'm just going quick, because we have very, short of time. So similarly, we produced another peptide and we looked at it. It is also working in the same way. We determine the molecular weight, we check the inhibition, we check the purity, single band, and it is inhibiting, right? And then we determine the molecular weight analysis, and then we have studied. This is a Mycobacterium tuberculosis, right? See, MTB RV strain, which causes TB. This we carry out in the BSL 3 level facility. And it is actually killing the Mycobacterium tuberculosis also, right? Interestingly, it actually goes inside the cells without causing any damage to the cells, right? In the lung, in the macrophages. And this is what is, macrophages do not call anything, right? But actually it kills this bacteria residing. This is a mice experiment that we actually carried out. Right. Similarly, we actually characterize some lengthy peptides. Story goes on. Right. When students, characterize each one peptide, then it is published, and they actually feel the credit like this. These are mice. Actually, how it is protecting, right? See, when this is a negative control, which is actually allowing viable bacterial count high, but when we are actually treating, it's coming low, right? It's a blank control. Nothing is there. Similarly, when we are using, this, injecting into the mice, it is actually protecting. You can see over here, right? So when we are, this is survival card, right? So it actually protecting the mice. These all studies are very essential, right? Mice studies, all this. Without this, they never allow, right, you to go into the market for therapeutic applications. This is very important. You must have heard, seen that in the pandemic, the vaccine, there was a lot of discussion going on in the media that they have not done in the clinical studies or clinical trials, etc., right? These are required before establishing that, right? It should not kill any, or it should not have any side effects. That all will determine initially cell lines, then animal studies, then monkey studies, then it will go to the clinical trials. There are various aspects that needs to be done before introducing to the market. So that's why it takes 10 to 15 years time. Similarly, another student published one more story, and this student actually determined a virgin molecule from the Virgi bacillus, and you can see over here there is no hemolysis, right? Cells are actually retained over here inside. Right? Similarly, we also characterize the lactobacillus species, and we have done the MALDI, and we observed that how exactly it can protect. We observed the bacteria, how it is getting lysed. And also we looked at the immunomodulator properties, etc., etc. So all this work go on in a flow, right? Once after the discovery of a bacteria, right, you can actually have many things, right, in the line, right? So that once you have done all these things, then it is called as a characterized peptide. And now this peptide, you can actually use it, for further studies or even go for the therapeutic application, right? I'm sorry that I have to be a bit quick, right? Because its time also is very important that speakers must be waiting. But you are most welcome to visit my lab. This is my lab at CSR Impact. These are my students, right? A while ago, but a couple of students already left, right? She's pursuing, and she has gone to US. She's actually going to some university, and is youngest one, although looking very tall. Right, so this is my lab, and this is our institute. We have a beautiful landscape where actually a DNA, flower artistic, this is a natural flowers. Actually, they're made like this in our landscape. Thank you very much. Yeah, Anu.
Thank you, sir. Karnakar sir. Karnakar sir. Hello.
Yeah, stop.
Karnakar sir.
Sir, I'm in online. Sir.
Yes. Please extend vote of thanks for keynote speaker, no?
Sir,
and the
sir, thank you,
concept
sir.
what you have elaborated in for this webinar.
Okay, sir. Thank you, Suresh, sir. You are a live example, sir. We arranged this topic. The reason is your belongs to Sadasupet. My students can inspire by seeing your lecture or your example. They can reach in a good position. Sir, you are explained very well the condition of the earth and biological evolutions and cultivation of microorganisms and instruments involved in characterization of microbes and extraction of peptides, sir. Especially shown the probiotic microorganisms also. Next time we will arrange your lecture, especially on probiotics. We expected, sir. Sir, thank you very much for spending your valuable time for this workshop. Thank you, sir. Thank you very much.
Thank you very much, Dr. Karnakar and Anuradha. Right? This is a very good initiative, and we can maybe sometime later I asked my staff, because of the COVID, we are unable to demonstrate certain things. Otherwise, actually, a live demonstration also we can actually make it possible, right? Okay. And students should get exposure, right? Then only they can actually motivate it to us. Almost my home example, I am saying, even our kids also not ready to take the science stream. They only prefer math or computer science. But trust me, life science, my student sitting over here, right? She's, aspiring, right, to go abroad and do some good research, right? She's from, far, mountaineer area of the Himachal Pradesh, or, she's from Leh, Ladakh. Right, and she actually want to pursue, thrive in science, actually. So people are there, actually. So we should encourage these students, right, to take the science stream, so that actually they can go, they can do some wonderful science research.
Thank
you
you,
very
sir.
much.
Thank you.
Yeah.
Can you express and show your mail ID, sir, if anybody want to interact?
I already sent it in the chat.
Okay, sir. Thank you, sir.
I already sent it. Okay,
Thank you very much, sir. Thank you.
Sure.
very much.
most welcome.
The next
Thank you
resource-
once again, Suresh sir, on behalf of our principal, teaching and non-teaching staff and the students, those who learn something with these type of webinars. And lastly, you mentioned there is a lot of scope in life sciences and also physical sciences, and it granted some students, when they scared about certain science subjects, they want to take mathematics or else computer science, even some of the students arts also. And once again, for enlightening the concept of this advanced technologies in biological and chemical sciences. Now I request Dr. Karunakar sir to introduce Professor G. Rambabu sir, and who are very near to us from Geetham University.
Thank you, sir.
Over to Dr. Karunakar sir.
It's my pleasure to give brief introduction about Dr. G. Rambabu garu. Dr. G. Rambabu garu presently working as professor in the Department of Chemistry, Geetham University, Hyderabad. Sir completed his PhD from Usmania University. After completion of his PhD, he did postdoctoral research from California University, USA. Sir worked as a research scientist in industry, also GVK Biopharma from 2008 to 14. Sir interested area is small molecules drug design and development. Sir published 80 publications in national and international journals. Sir have 3 patents in the area of medicinal chemistry, molecular docking, pharmacophore generation. Sir completed 2 major projects sponsored by DST. Under his guidance, 7 PhD students completed their PhD. 15 students are doing research under his supervision. Sir received Best Scientist Award from GVK Biopharma in 2012, and also Best Researcher Award from Vice President Venkaiah Naidu from Geetham University. Sir has a friendly nature, very cooperative. I am very happy to invite Professor G. Rambabu garu. Sir, I request you, please deliver your topic in this workshop. Sir, Rambabu sir, please. Just sorry. Just a minute, please. Sir Jan will within 1 minute. Actually, we allotted time 11:30.
Hello,
Sir.
sir, can you hear me?
Yes, audible, sir, audible. Sir,
Okay, okay.
You can proceed, sir. Thank you, sir. Thank you.
Thank you. I mean, I am on time, almost.
Yes.
Huh?
Yes, sir, we are advanced.
Sir, please give me host
Okay.
permission.
Okay, sir.
I will share my presentation.
Bhavani?
Just now we have completed my
Yes,
my
sir.
scholars Vaivava, sir.
Yes, sir. Congrats, sir.
Thank you, thank you. Can I share?
Sir, in which name you are joined?
Yeah, fine.
Okay.
Rambabu.
Okay, sir.
Yes, sir. You can see it now.
Yes.
Okay, shall I start, sir? Karnakar Garu?
Sure, sir. Sure, sir. Sure. Please.
Yeah, please. So now it is 11:30. How much time I can take, sir?
Up to 12:30.
Okay, 1 hour. Okay, good morning, everyone. I would like to start the drug discovery program, which explain, especially which will align with the current day research. That is, we are the people who are attending from home, and lecturers also are delivering from home because of these viral problems. So that is the reason. Just I have taken what is the way of developing or discovery the drugs for the drugs, especially correlating with the viral diseases. So today my topic is, why are antiviral drugs so hard to develop? Okay. So first of all, what the procedure I am going to explain, the drug discovery and development, the procedure and the timelines, success rate, etc. And then we will try to take who are the people causing the problem, that is a disease, we ourselves causing the problem, or any pathogens are causing the problem, who is the main culprit, that I am going to discuss, and identifying the problem and who is the causing the problem, how to control the problem by arresting the culprit. This is what my main today's discussion. So you can see it here. You can see it here. Sorry. You can see it here. This is the virus, and we need to give the drug to kill it. Of course, there are so many drugs. Okay, our body, I mean, now if you see, our body is with the full of drugs. Without a drug, I think we cannot move ahead in the life. So of course, Dr. Karnakar might have introduced myself because that time I was a little busy with some other. So myself, Dr. Rambhava, and Professor, Department of Chemistry, School of Science, Gitam, Hyderabad. This is what my campus, which is located Rudraram, Patancheru Mandal, Telangana, near to your Sadashivpet. Okay, we are having almost 250 acres land here and very big building. And whoever wants to visit our company, our university, please, you are welcome to our university. And whoever wants to use even research facility in our university or wants to have a collaboration with me or with the Department of Chemistry, you people are well. I mean, we are welcoming you all. Let's go to our topic now. This is the time to define what is a drug. First of all, we should know what is the meaning of drug. Drug is, after all, small molecule. We know nowadays everyone, I think, at everyone's house there is a Dolo 650. Okay, what exactly it is? It is a very small molecule, parasite. Who discovered it? Proudly we can say we. We means chemist. Who are the people who has given the birth to the paracetamol? That is what the drug. We are the one who synthesized in the laboratory. Chemist, I mean. Okay. Okay. Now it is ruling the entire country and saving the entire world people's life. So this is a simple definition. Natural or synthetic molecule which is used for the treatment or prevent of the disease or giving the relief from the discomfort. Okay. So it is a counterpart to the disease. It is a counterpart to the disease or giving the relief from the discomfort. Here, all these compounds either being synthesized by ourselves or extracting from the natural sources. You can see it here. Some of them are natural products we extracted and tested against the disease and discovered as a drugs. And some of them are we ourselves synthesized using our chemistry knowledge and tested against the different diseases discovered as a drugs. Now, as I told you, drug is counterpart of the disease. Drug is counterpart of the disease. Counterpart of the disease. 1 minute. So now I want to define what is a disease is. Disease is actually simple, we say pathological state or abnormal state. Okay, normal state as a physiological state. I am very fine, comfortable, healthy. That is physiological state. Pathological state. I am suffering with the fever. I am suffering with the cold, cough, pains, etc. Suffering with cancer, AIDS, etc. All these are the disease conditions, which we call it as a pathological state, which is affecting the organs or which is affecting the body. Where it will start, how it will start disease. Okay, disease starts with, of course, changes of the biological system. Okay, it triggers from the protein or gene level or organ level and affecting body level. So here, the simple definition, mostly one can say that in our body, there are, diseases caused by ourselves and pathogens. And I will come back exact definition of the disease. Who is causing this disease? Okay, the simple you can see infection diseases. Okay, and the second one, non-infectious diseases. Okay, bacteria, virus, germs, fungi, etc. are the ones which are caused by the pathogens. Non-infected, they are the infectious. Non-infected, our body itself, we ourselves create the problem. The others, pathogens are creating the problem. Bacteria are creating the problem. Viruses are creating the problem. Germs are creating the problem. Of course, they are not having any relation. The other people, okay, definitely they may cause the problem. There is no wonder. But why our body itself is causing the problem? Why our body is enemy to us? Okay, that is non-infectious diseases. So here they are the communicable, non-communicable, again, two type of diseases. Non-communicable, all pathogens related, viral, bacterial infections, etc. are the communicable, and our body itself causing the problem like cancer, BP, diabetic, or Alzheimer's, Parkinson, different diseases, heart, stroke, etc. They are caused by our body itself. They are the non-communicable. That is with the ME1, non-communicable diseases. You can see it here. Okay. Cancer, chronic lung diseases, diabetic, heart diseases, stroke, all these are due to why our body itself is causing the problem. Because our habits, okay, our habits, our food, our environment, these are the ones which are influencing our body, and our body is prone to different diseases. Physical inactivity, alcohol abuse, tobacco, unhealthy diet. These are the four major factors which are really causing problem to our body. Okay, so I mean, these are the four. If you avoid, okay, obviously we will be healthy people. Okay, apart from that, of course, that is related to our, I mean, activity. But even then, we are having of the good diet. We are taking the good diet, non-alcoholic, and doing the physical activities, not even touching the tobacco, but still there is a chance to prone the different diseases which are caused by the pathogens. So here you can see some of them. The bacteria are viral related diseases, malaria falciparum, Plasmodium falciparum, and viral major, and cholera, AIDS, anthrax, corona. Okay. Now you are seeing so many dengue, okay, typhoid, all these are, okay, the infectious diseases. Here you can see some of them. Malaria, E. coli, single-celled organism without chemical. Okay, throat infection. Food caused by the different pathogens. Now we know broadly, I mean, two people are causing the problem. One is ourselves, second one is others. Then this is a problem, of course, it is associated, but these people and get the different diseases. Then we need to have an alternate. Okay, alternate is, okay, drug. So we need to identify the drugs for the different diseases. Okay, so how it starts? What is the meaning of drug discovery is identify suitable molecules for a disease. Of course, as a synthetic chemist, we know that we are preparing the different compounds or extracting the different small molecules from the natural sources. Our job is coming to the laboratory and cooking the different molecules, characterizing them and saying that this is what the compound. This compound can be synthesized, that route, this route, etc. Or as a synthetic organic chemist, our job is synthesize the compound with the different approaches or different methods. One step ahead, we should become the medicinal chemist. What is a medicinal chemist and what is the synthetic chemist? Okay, we need to synthesize the compound by keeping in mind that what is the or how this compound is going to be medical important, how it will have the significance of the medicine, I mean medical property. Okay, so this is what we are bringing medicinal importance to the compound is nothing but of the drug discovery. Finally, this compound can become the drug which we are synthesizing today. Of course, if you see the paracetamol, very small compound, now you see how it is saving our lives as well as generating the revenue or whatever it is. And it is a blockbuster drug now. It is, if you see that, it is having the 3-4 steps to synthesis and very small molecule it is. Okay, fishing out the suitable molecule is nothing but of discovery of the drugs from the bunch of the compounds or bunch of the molecules. Now, of course, the drug discovery starts with the disease. First, the drug discovery starts with the disease. Okay, disease, everything, disease, who is causing the disease, what is the disease, what is the protein, what is the target, what is the gene, what are the symptoms, everything this will be done, research, this type of research will be done by biology people. Of course, what biology, microbiology, biochemistry, all these genetics, whatever it is, all this will come under the biology umbrella. So what we say is the biology people will try to diagnose what exactly the disease is. And they are the one who is saying what protein or what gene causing the problem. Okay, so as I told you, our body is with the full of proteins or enzymes. Each protein is having their own function. Function is nothing but of work in our body. There are n number of proteins. Each one is having its own function. Okay, when the disease is caused, over functioning or under functioning of the protein causes the disease. That is what our enzymes are proteins. Over functioning of our protein or under functioning of our protein causes the disease. Or pathogens which are into our body causes the problem. These are the two major ones. So biology people will come out what is the protein or which virus or which bacteria causing the problem. That is the number one. Here you can say entire biology people will work on it. Identify disease state and relevant biomolecular targets. These are targets are nothing but of enzymes or proteins or RNA or DNA, etc. Now, this is the parallel, apart from this, parallel to this, we are the ones chemists. Okay, we are the ones who is keep on synthesizing the different compounds using of the different synthetic approaches or methods or schemes, and apart from that, extracting of the different compounds from the natural sources. This is the chemistry. This is the chemistry here, and this is the biology. Okay, these are two people now, they don't have any relation. These people are doing their own job. These people are doing their own job. Okay, and he is the one who is suggesting, hey, this is the protein or this is the enzyme which is causing the problem. Now the chemist, I am saying that, hey, I am having these 20 or 30 compounds or 2000 compounds. Now actually if you see, there are 100 million compounds. 100 million compounds. They are in the stock. Different compounds if you start with the benzene. I don't know who is a suitable person to this target, because this is the one which is causing the problem. What problem? Either over functioning or under functioning of this protein causes the disease. Then this is the one which is a counterpart. This is a drug is a counterpart, and will say that, hey, you are doing the overaction, you come to the normal action. This is the one which will tell to that enzyme by binding each other. The protein or enzyme and the drug bind each other and try to bring back over functioning or under functioning of the protein or enzyme to the normal function. Okay, now I am taking this protein. Sorry. Now I am taking this protein and this small molecule, and I am testing using of this in vitro facility. That is a primary assay. In vitro means in the laboratory. Simple. This is the enzyme, let us take, and this is the molecule. P plus L. This is the protein plus L is a ligand. Okay, P plus L. The simple chemistry we are making and trying to form the complex. Then this complex, not all the chemicals will form the product. As we know, A plus B is giving the C. Not all A's will bind with the B and form the C. Only whichever suitable A and suitable B only will form the C. So here I am having this number of compounds, and this is what the protein. Now I will be taking this protein. I am taking one by one compound. They are the ligands. Ligand 1, ligand 2, ligand 3, ligand 4, ligand 5. Like that, how many compounds are there? 100 million compounds are there. Okay. I will try to test it and will try to find which are the suitable compounds. Here I am telling suitable compound. That compounds, now I am calling it as a hit. The compound which is having the minimum activity on the target protein which is causing the disease, that compound I am calling it as a hit compound. Now I am taking this hit compound and trying to test onto the rat. Here you can see this rat. Okay, here this is a in vitro study. Now in vivo study. In the animals I am checking, that is a secondary assay. Okay, the compounds which are promising at this level will be subjected to test onto the animals. Now I am checking onto the animals and trying to see how the animal how these compounds activity in the animals, because in the animal this enzyme or protein is there. Now our target is that protein and enzyme only, but when it is there in the living system, that is animal. Now same drug will be injected onto the animal and trying to see how it is reaching the target protein and how it is binding in the living system. Then we will try to see the efficacy of the compound and ADME properties, absorption, distribution, metabolism, excretion onto the rat and toxic effects. So once this compound shows the promising results at the in vivo level or preclinical level or animal level, those compounds will be subjected for the clinical level. Clinical level is nothing but onto the human beings. Onto the human beings. Now we are seeing in the clinical level, there are 4 phases in the clinical level onto the human beings. One with the onto the healthy volunteers. Why we are seeing the healthy volunteers? Because already I tested this compound onto the rat, and the rat it is very, very safe. It is not causing any problem. Suppose. Then if I test that compound onto the human being also, there will not be any problem, because we are having the 80 or 90% sequence, genetic sequence similarity between animals and us. Therefore, if it is not working or if it is not causing any problem to the animal, we are also animal category. Definitely, it will not cause any problem to this animal also. This animal means us. Therefore, I am taking it granted that this compound is not going to show any toxic effects onto the human beings. We will start with onto the healthy volunteers, and we will try to see on the healthy volunteers how the ADME properties are: absorption, distribution, metabolism, excretion, and toxic related side effects, etc. Because directly I cannot test onto the patients, because the patients are having the less immunity. If I test onto the patients, it may cause some other problem. That's the reason we would test onto the healthy volunteers and make sure that it is not causing any problem, or it is having the good ADME property. Then we will try to take the phase 2. That phase 2 is nothing but onto the patients. Now, the safety is measured using of the phase 1. Now, phase 2, I am taking the patients and trying to see the efficacy. Efficacy means how much time it is taking to reduce the disease. Okay, what are the even problems onto the patient? ADME and efficacy will be studied. Then we will try to see how much time it is taking, how much dose we need to give, etc., etc. Clinical trials will be carried out in the phase 1, phase 2, phase 1, and then if it shows promising results in the phase 1 and phase 2, will be subjected for phase 3. Phase 3 is nothing but of making more robustness. Means this drug will be subjected for the more number of volunteers and with the different conditions at the different country people. And I mean, it's like changing, because if it works on Indians, okay, there will be a change onto the Americans or Chinese, etc., European. That is the reason to make more generalized, the compound will be subjected for the more population. Okay, and if it shows promising results at the phase 3 also, phase 4 will be the continuous process until unless the drug is really, withdrawn from the market. Phase 4 is continuous as long as the drug is there in the market. Again, testing onto the human beings day to day and trying to see, any changes is happening day by day. That is a phase 4 continuous trials. After getting of this phase 3 approval, okay, people will file it as a new drug application and will apply for the FDA for the drug approval, Food and Drug Administration. This is a Food and Drug Administration, US, or here ICMR in our India, and European Medical Agency from the Europe, and like that, each country is having their own medical agency where they will go through the entire research documents and will try to find whether the data is up to the mark or is there any problems, and sometimes they may ask us to reiterate some experiments and produce the data. Until unless they are being convinced with our data, they will not giving the approval to release as a drug. Once you get the approval from the different agencies, you can release it as a drug in the market. This is what the total drug discovery process. Okay, nowadays people are, trying to see, okay, the computer application also. As I told you, there are millions of compounds, and rather before going to the in vitro studies, people will try to take the computer-based studies. Like this is the faster technology where it will mimic the in vitro studies and take the protein in the computer and these small molecules. They will try to calculate the free energy using of the thermodynamic properties as well as the quantum chemistry. It will calculate the spontaneity of the small molecule onto the protein and will suggest few compounds to test using of the in vitro facility. That is computer-aided drug discovery. So this is what total story here. This is a, drug discovery pipeline. You can see it here, target discovery. Okay, here we are, strategic research. What is the target? Target is nothing but a protein, where we are identifying the hit and the lead compounds. This is what the chemistry, virtual screening, high throughput screening, drug designing, hit to lead compound. This is a early drug discovery up to here. Advanced drug discovery will start at the preclinical level, as I told you, okay, testing onto the animals, and fully developed compound onto the clinical level, and FDA and EMA review and post approval. This is the phase 1, 2, 3. This is what I have explained. This at this stage, after the clinical IND, investigated new drug. Here, after clinical, that is a NDA, new drug application. So after going through entire this process only, after getting approval only, we can say that compound is a drug. Up to that, we cannot say it is a drug. people generally use drug, one should not use. That is the final product. Okay. Now, this is what the success rate, cost, and time we need to see before, because this is the one of the major problem in the drug discovery. What exactly? The success rate. You can see it. Suppose I started one drug discovery program. My objective is to discover a drug against particular target protein, which is causing the disease. So now I am starting with the 10,000 compounds, let us assume. Okay, here, as I told you, 10,000 compound in vitro studies in the laboratory where isolated protein on these compounds will be tested. Nowadays, people are using high throughput screening. Okay, after that, second one is using of the animals, and the third one is a phase, clinical trials. So suppose if you take 10,000 as the initial hits, to come out with the preclinical active compound, it will take 6.5 years time. So preclinical level means animal level, only 250 compounds, the remaining will be rejected. They are not showing any activity. 250 compounds only passing the preclinical if you test 100 compounds. This is also probable data, not exact data. Okay. Now let us assume 250 compounds are passed and shown the good activity at the preclinical. Now these 250 compounds will be subjected for the further study, that is the clinical trials. These 250 compounds, if you do the clinical trials, there is a chance of getting 5 compounds. Okay, these entire 5 compounds data, if you subject for the FDA, there is a chance of getting 1 compound as a drug approval. This is what the probability. If you start your program with the 10,000 compounds, you are ending with the 1 drug. This is what we call it as early drug discovery. This is what the preclinical. This is what the clinical, and this is what FDA approval. Here, 10,000 to 1 compound. How much time it is taking? 6.5, 12 years, and 1.5. That is 7, 8, 14 years. 10 to 15 years of time it will take. If you start today, that means 22, there is a chance of getting as a drug 1 compound by 2030. Now you can see how much 10 to 14 years you need to wait to know 1 compound as a drug if you start with the 10,000 compound. Okay. Apart from that, the success rate you can see 10,000 out of and 1 compound means 10,000 to 1 compound. The success rate is 0.01. Now, how much money I need to spend? $800 million approximately. 800 million means 1 million means 10 lakh. 800 into 10 lakh. Our currency, if you take, that is, let us take 70 rupees average. Okay, 800 into 10 lakhs into 70 rupees. So approximately, you can see the, around that much money, around almost 40 crores we need to spend. Very huge amount we need to spend because there are so many departments, so many people, so much of expenditure is required. That is the reason it is costing a lot. So success rate is very, very low. Now question is whether we can take up this or not, whether we can take up this program or this project or not. Because very, very less success rate, too much time it is taking, it is very costly affair. So that is the reason no businessman will take this project, because I cannot wait 14 years. Okay. And to get the 0.01 success rate and need to spend more money. So if I spend this money within 2-3 years on any contract, okay, within 3-4 years, I can get my money back with the double amount. That is the reason India like companies or business magnates or people, they are not showing much interest. They are not showing much interest to investigate onto the drug discovery. That is the reason we are lagging or we are behind, somewhat lagging behind the research or discovery of the drugs. Hardly there are 1-2 compounds which were discovered by the India. Entire, you can see it. Of course, one can say our India is having so many pharma companies, so much of research institutes, everyone. These are almost service, and almost we are doing to get the good publications on patent. Till date, there is a, a very good product which is turned out as a drug and helping the society. That's what actual our problem. Of course, I am going to discuss that in future. The simplest drug mechanism one can understand, the lock and key mechanism. Okay. Key is nothing but of the final drug, which will work on to the lock and try to open or close. That is nothing but of drug. Okay, there are so many keys, but all keys never work. May not work onto this lock. Only suitable key will work. This is what the simple, understanding of the drug discovery is. Identify the suitable key against the lock. Lock is the one which is causing the problem. Okay, and there are, as a businessman, I'm saying, I want to spend money, I have money, and even I'm not bothering about the success rate. But can you reduce the time? This is what even all the people are asking. I don't want to wait 14 years time to know whether I am getting the success or not. 14 years is too long. That is the reason people started thinking of how can I reduce the drug discovery program time. Then these people are trying to implement advanced technologies like computer applications, etc. One of the computer applications is computer-aided drug discovery study, which is a faster technology. Of course, as we know as a chemist, Heisenberg uncertainty theory tells, okay, accuracy, if you start 2 things at a time, there is a error. If you start 2 things, there is a error in one activity, there is a accuracy in the second activity. So similarly here also, it is very faster technology, but success rate is very, very less comparatively exponential. Of course, one of the computer application is the structure-based drug design study, where people will try to take the enzyme in the computer and will try to see 3-dimensional manner, will try to see what is the active site of the protein and try to design the compound. This is what structure-based rational approach designing of compound, fulfilling the interactions between the protein and the ligand. So using of this, as a chemist, we know the thermodynamic parameter. Okay, protein plus ligand is giving the PL complex. Now I can calculate the free energy delta G equal to delta H minus T delta S. Where I can say enthalpy and entropy is taken, will be calculated delta G value. Delta G, if it is a negative, we know it is a spontaneous process. If delta G is a positive, it is a non-spontaneous, those compounds will not be taken for further testing. So this is what people will start taking of the different chemical structures, filtering down, and trying to identify the suitable compounds as a drug against the disease. Now coming to our story, that is nothing but of, common viral diseases. As I told you, what are the challenges to discover the viral drugs? Okay, so different viruses, you can see different shapes they are having. Okay, coronavirus shape, you can see rhinovirus. Okay, herpes, hepatitis B also it is virus. Rotavirus, mumps, measles, HIV, HPV, smallpox, rabies, norovirus. These are the different viruses which are, penetrating into our system and trying to cause, damaging our body, causing the problem. Okay. Now I am going to discuss one by one and what is the symptoms and the treatment, etc. and the drugs also. You can take this one. Okay, route of viral transmission. This is you can see the simple mechanism how it is. This is a virus. Okay, it will penetrate into our host cell. how it is coming. Okay, after coming into its cell, it will release the genetic material, entire our enzymes, okay, penetration, attachment. First it is attachment. Our host cell is giving our protein targets, are hosting this cell to penetrate into. It is a penetrate, it is the nucleus, and it penetrate into our cell. This is our cell, host cell. Then it releases its genetic material. Viral content are released. Then biosynthesis, our body itself is helping to grow this virus. Then it is making the budding. Previously, only one virus entered. Now it is started growing. And once it grows, and again it will come our cell and release into our body. Again, this multiplication can happen and will try to produce. That is the reason overnight the lakhs of the viruses will be grown in our body, because it is cyclic process, and one will, for example, one will produce the viruses, three, that is 9 into 3, like that proportionately it will, the viruses will grow in our body. Okay, so what are the viral transmitters? Skin contact, respiratory, nasal, oral, milk, transplantation, sexual, insect vector, animal bite. These are the ones which are helping to transfer the virus into our body or our cell. This is what the pipeline, if you see, evaluation of the antiviral drugs, it started 1960, and now we are here 1920 with the corona, where the latest one is the favipiravir or remdesivir, which are treating for the coronavirus. Okay, there are so many corona, virus-related problems and discovered the drugs. You can see it. This is what the flow diagram. I don't want to explain entire this. HIV first case and a therapy developed in, and still we need to develop a drug which can be useful for the HIV treatment. Still there is no proper drug which can treat. Ritonavir, all these are, if you see it, the compound names here, it will end with VIR. VIR, that is ritonavir, remdesivir. Okay, you can see all these names. Favipiravir, VIR, that is virus. VIR indicates viral drugs they are. Coming to the different diseases here, smallpox. Okay, this is what the you can see. This is what the virus. This is what the virus, poxvirus, variola poxvirus. This is which is causing this smallpox. Mode of transmission by air, and the symptoms: small pinkish itchy spots appear on the body with the fever. This is a tecovirimat. This is what the drug which is using to treat this one. Similarly, chicken pox. You can see it here. Varicella zoster. This is the pathogen virus name. This is mode of transmission. Again, for the direct skin contact, and the symptoms, you can see this type of, small spots, and with the body ache, fever, etc. And this is acyclovir. This is what the drug which is used to treat the chicken pox. Measles. Okay, here rubella virus, transmit droplet infection. Droplet infection, you can see how it looks. Okay, and a red spot, fever and swelling. Almost all viral or bacterial, immediately you will get a fever. What is the meaning of fever? Fever is not a disease. Fever is a symptom. Symptom, what it tells. That means, hey, your body is attacked by some X person. Be careful. So, in our body, one person entered. We are not getting the fever immediately. After entering, incubation period is there. 2-3 days it will take. After 2-3 days, there is a huge amount of the growth of the viruses, lakhs. All these are trying to produce, because our body, previously there is a space, empty place. Now it is 1 lakh people inside our body. Then all these 1 lakh people are colliding each other and producing the temperature. That is what our body is getting full of temperature, indicating that our body with the more number of pathogens. Then we need, our body is cautioning, I need treatment. Then immediately we need to do the treatment for the particular disease. And even polio, you can see that is also polio virus name itself. And these are now, almost people are, we got the vaccine. So almost all these polio related problems were, arrested. Almost we are giving treatment. Rabies, again here it is, biting by dog, cat or monkey. This is a rhabdovirus, that is, which is a passing, and you can see it here. By the bite of the this dog, this through this nerve, it is attacking our brain. That is the reason very, very dangerous this rabies virus is. Immediately we need to treat, because it is simply attacking the brain nerve system. Okay, and the sixth one is influenza. Okay, this is what the virus name, influenza virus, and droplet transmission. And the symptoms you can see in anywhere there is a pain or headache or fever are the symptoms. And the treatment includes the synthetic drug amantadine, which blocks viral penetration to the cell. Then mumps also, because of the virus only, one will cause this problem. Mumps virus, that is a paramyxin virus here. Droplets, direct contact with the skin. Okay, again, gland of the salivary gland. Okay, infection in the salivary gland. Okay, and getting of the fever, and of course some pains. Prevention is, okay, vaccine. We are getting the vaccine. Okay, there are no drugs. And the other one, HIV, AIDS. HIV is a virus and AIDS is a disease. Please try to remember, HIV is a virus, AIDS is a disease. There are, here you can see the right side, there are n number of drugs which are using to treat the HIV, AIDS. Okay, these are the drugs, and where people trying to identify using the computer-based drug discovery, you can see some of, these are drugs here which are nucleoside and non-nucleoside based drugs. Hepatitis, this is also the viral causing disease only. There are A, B, C, D, E, hepatitis virus A, B, C, D, and this is what, hepatitis jaundice. This jaundice generally is caused, okay, due to the water infection. Through water, this virus will, enter into our body. This is the jaundice that is caused by the hepatitis A. It is not a problem one, but hepatitis C, very dangerous one. Okay, hepatitis C and B, these are the very dangerous one. Hepatitis, okay, A, B, D, E, there are vaccines. C, still we need to get the vaccine. And hepatitis B, C, D, blood, sperm, and other body fluids through, okay, it will penetrate into the or transmit one person to the another person. And hepatitis A and E, lack of food hygiene and contamination of water and substandard sanitary facility. Okay, it will cause this. And these are generally, this is a liver. So if the person gets the hepatitis related problem, that means there is a damage at the liver. So liver, it is a very prominent organ. Therefore, we need to be very, very careful. Liver related disease, liver and the lung. Now coming to the dengue fever. Okay, it is actually mosquito-borne disease. Of course, mosquito is a mediator between the host and guest. But guest is Aedes aegypti mosquitoes. They are passing through mosquito bite. Okay, here you can see, this is a mosquito which is uninfected, but this person is having the dengue. Then it is having full of this blood, which is sucked from this person and will transmit to these healthy people. These are the symptoms: headache, pain, rashes, etc. And the current status of vaccine and the drug for the dengue. Dengvaxia, a Mexico-based pharma company discovered but Sanofi that is, but the problem is it is not approved because it is causing many problems. Therefore, it is not released this vaccine to treat to protect from the dengue virus. Okay, to date, there are no drugs. Here, some of the drugs, chloroquine and this Balapavir, NITD, these are the 4 or 5 drugs which are in the clinical trials, not yet confirmed. To treat this, of course, to control fever, is the paracetamol. Of course, it is a universal drug. And apart from that, there are some natural products, that is Caripil, that is papaya extract, which is approved by the Ayush in 2015. Of course, these are the drugs which are identified as the drug repurposing purpose. I mean, using of the drug repurposing method. I will try to explain what is the drug repurposing is. Now coming to the current disease, that is COVID-19. Okay, coronavirus. It is the disease name is COVID-19, but it is caused by the coronavirus. Okay, of course, we know already almost all people who got infected, that we know the symptoms and the treatment also. I mean, now we are having the vaccine, okay, that is Covishield and Covaxin, etc. But it is directly this virus affecting the lungs. Okay, drug repurposing, or this is the method which is being used to get the quick drugs. Using of the drug repurposing only, this is a remdesivir and a favipiravir and identify as the drugs. Okay, so here discovery, this is what the traditional discovery. Now already there are some drugs. We are taking the drugs. These drugs directly may not be acting onto the current disease. They are drugs for some other disease. We are rerouting it. That is what drug repurposing. They are the drugs which are for some other purpose. Now we are taking them and rerouting that and trying to see whether this can become the drug for the current disease. For example, I will explain in the next slide. Hydroxychloroquine. This is what the drug which is used for the treatment of the malaria. Now this is rerouted to treat the corona. Okay, initial days. Okay, this hydroxychloroquine, that is HCQ, was used to treat the corona disease if it is very dangerous condition of the patient. Similarly, favipiravir, here you can see the favipiravir drug. This is a drug repurposed approach people applied. That is previously it was Ebola drug. Of course, now also it is Ebola drug, but now it is rerouted to treat the COVID-19. This is a favipiravir. Come to the remdesivir. This is a treatment. This is used for the treatment of the hepatitis, but now people rerouted to treat the COVID-19 because these are two sequence. The viruses will have the more sequence similarity. Therefore, if it works onto the virus, there is more chances to work onto the other viruses also. That is the reason people will try to see, test the drugs of the different virus onto the new viruses also. Remdesivir, this is what the mechanism of the remdesivir. This is what the coronavirus, okay, which is our ACE2. This is what our protein, okay, host protein, our protein, which is accommodating this protein by interacting with protein interactions, penetrating into the system and releasing the viral RNA. And where this is one of the enzyme, RNA dependent RNA polymerase viral enzyme, which will convert RNA into the RNA into coating one and making of the more RNAs, viral RNAs, and will be budding the new viruses. This is one target, very important one, and this is the ACE2 is another target protein. Either we can block it here if we identify the suitable drugs, or we can block it here if we identify suitable drugs. Okay, so if you identify the drug will come first and will try to sit it here, will not give the scope to interact this protein. Therefore, this is a cycling process will be stopped. Or you can target this RNA dependent RNA polymerase RDRP, one of the enzyme which is crucial for the survival of the virus. You can stop it here so that the viral multiplication can be stopped. Common drugs for the viral diseases, of course, people generally will try to use the cough syrup, cetirizine and paracetamol. These are the three drugs people trying to, it is just controlling only. None of these are acting onto the viruses. Okay. Why are antiviral drugs so hard to develop? Because the viruses is there in our body. Okay. So whatever this, let us assume you need to shoot the virus, but you should not harm the host. How can I do? That person is sitting inside, not outside. That is what bigger challenge. How to stop the virus without damaging the inner working of the healthy cells. That is a challenging job. And the second one, biggest, drugs should not hurt the human host. Viruses are much trickier targets than the bacteria. That is what even we can treat the bacterial diseases, but unable to treat the viral diseases because viruses are more tricky than the bacteria. Viruses changes its shape on genetical level. That is what mutations. That's what people are saying. That variant, this variant. That is nothing but of. Mutations can occur in the genetic level. Okay. Given the drug discovery development is an inherently lengthy and costly undertaking. Antibiotic abound, but virus fighting drugs are harder to come by. The limited funding agency for the academic research projects. Always we see the commercial point, but we need to see the research point also. These are the very neglected diseases sometimes. Okay, but people will not do the more focus. People generally will try to see which are the very familiar diseases and will pump the funding to it only. They will not focus on to the neglected or rare diseases. That is also important. And we need to forecast what could be the future problems and need to work accordingly. That is what the research means. Developing an antiviral to treat emerging viral diseases will require consideration of new consortium models, including public-private partnership. So this is what lack in India-like countries. Okay, where the private people, they do their job and try to, academicians, whatever they know, they do and they will try to publish it, but there is no, collaboration or interaction between these two people and not, unable to work for the objective of our research. We are not focusedly working to, solve the problem. Independently, we are working. Prevention is better than cure. Therefore, having of the good food and taking care of the ourselves, wearing mask. These are the ones every day we are hearing. I don't want to repeat it here. So that is the reason it's a better, prevention is better than cure. So this is what overall my talk. I think, what is my time? Yeah, 12:27. I am at.
Shall we ask?
Now you can ask me if you have any questions or clarifications.
Sir,
Thank you.
Sir.
Sir.
Please, what are the minimum requirements for doing computer-based drug design?
Minimum requirement means there are some softwares, sir, drug discovery softwares. Okay. We need to, have a high-end PC with the graph card. And apart from that, we need to buy the software, which is costing more.
Is there any free available software?
So in your Autodoc is there, sir. Autodoc, one of the. There are some free software, sir, but anyhow, free software, the results, the reproducibility of the results is, somewhat low when you compare with the commercial products.
Sir, how to get the protein part for docking? So
Protein?
part for docking.
Oh, protein path docking. Sir, that actually we, before, going for the docking, we need to understand the protein function, path, everything. Then we need to see and which proteins are the drug targetable ones, and those targets we need to choose. Some literature information and experience is required.
Sir, previously the speaker said, sir, there is a microbial bank is available in, is there any protein bank available?
Yes, sir. Yes, sir. Yes, sir. The protein banks are there.
How to get?
3D structure protein bank, that is PDB data bank is there. For the sequence also there is a data bank. Different data banks are there, sir. If you take the bioinformatics course, they will be explaining different databases and what is the use of the databases. Even genetic sequence analysis also is there, sir. NGS, next generation sequence analysis, etc. And for everything there is a database, sir, now.
Thank you, sir. I request Srimati Anuradha Madam, please propose the vote of thanks.
Thank you, sir. Thank you for giving me this opportunity. Good afternoon, everybody. It is immense pleasure to propose vote of thanks to eminent speaker and resource person of this session, Professor G. Rambabu Garu from Geetam University, Hyderabad. Sir has delivered a valuable speech on antiviral drug development. During this corona pandemic era, I think this is the most important topic to be discussed. A simple chemical molecule, either it may be in the form of tablet or it may be in the form of injection medicine, is a solution of most dangerous disease which is caused by a pathogen. Sir has explained about the drug discovery process, and he has discussed about drugs for different viral diseases also, and also about drug reappropriation. Sir's speech has imparted a great knowledge about the topic. On behalf of organizing committee of this online workshop and participants, we are very much thankful to you, sir, for choosing a great topic, a right topic at right time. Thank you very much. Now, over to Vinayak Kumar, sir.
Okay, madam.
continue.
Thank you, madam. Once again, I extend my regards and gratitude to G. Rambabu, Professor G. Rambabu, sir, from Geetam University. And sir gave elaborate causes and effects and the remedies for the so-called types of diseases. And that is what Madam, as Anuradha Madam already told, the relevant topic to the present situation, what the people are facing nowadays. And now I would like to introduce Dr. D. Ramarao, sir, from Kakinada. Sir, once again, I welcome you, Ramarao, sir, to this workshop and being organized by our college, that is Government Degree College, Sadasipet, by Dr. Karnakar, sir, and Anuradha Madam, in collaboration, and the topic which we already know, advanced technologies in biological and chemical science. Ramarao sir completed his MSc, B.Ed, MPhil, PhD, and working as associate professor and the principal for PR Government Degree College, Autonomous, Kakinada. And sir has 20 years of teaching experience and 8 years of research experience. Sir participated and organized many NSS programs and also coordinated Red Ribbon Club. And sir associated with the Rotaract Association and many cultural activities. And sir has 2 research projects for his credit, that is DAEBRNS, which worth of rupees 42 lakhs. And sir has 45 research publications and submitted papers, international 25 papers and a national 20 papers. And sir received awards many from other organizations. And sir also life member for Life Member Indian Science Congress, ACT Mumbai, and Indian Red Cross Society and Alliance Club International. Sir, we are once again welcome you for this national workshop and thank you for being with us through this virtual mode. Over to D. Ramarao, sir. Thank you.
Thank you, sir.
Bhavani, give host.
ఇచ్చేసాను సార్.
Okay.
Kanaka, this screen is okay? Karuna? My screen is available?
Yes, sir.
Okay?
Okay, sir.
Okay, okay, I will start. Thank you, Karunakar, for giving the wonderful opportunity, the one-day workshop. My friend Karunakar, my classmate, thank you for the giving opportunity. Okay, I am discuss about actually, I am carried out one important Government of India, DAE, BRNS, Department of Atomic Energy, Bombay, have carried out one major project. The title of the major project is the evaluation of groundwater and surface water quality, sustain the status in the NTP National Power Project. There is the Government of India planning for the establishment of the 100 megawatt power plant in Srikakulam district. That's why any power plant is established before the environmental survey and the existing environment status is measured in 4 directions. That is the soil status and groundwater status and air status is the main important key role, because after establishment of the nuclear power plant, the levels of the contamination in groundwater and soil and air, how much affected in the power plant. That's why we are carried out the groundwater and surface water. This is the part of our project, groundwater evaluation. So the establishment of the baseline data in the naturally occurring radioactivities in a Kovada, Srikakulam district. Actually, my project, the presentation is a very big presentation, but short and half an hour duration. Important headlines only I will explain. If any time I will explain in your college. We are planning also some demonstration methods also planning, but the COVID effects in this time we are not the demonstration, only explain the only explanation only. This is the scheme of the our topic and our project proposal. There is a groundwater, surface water is carried out. This is the parameters. Number 1 are radiological parameters, nucleides, uranium nucleides, chemical parameters, THEC. This is the TDS ORT, means oxidation reduction potential. The oxidation reduction potential also influence the quality of the groundwater. Temperature, pH means total hardness, calcium hardness, magnesium hardness. The ions, chloride, fluoride, total alkalinity, DO, dissolved oxygen demand, BOD, and COD, NO3, nitrate, phosphate, sulfate. These are the chemical parameters. And the stable elements, potassium, sodium, potassium, calcium, magnesium, and biological parameters also were carried out. Totally radiological parameters 5, chemical parameters 18, and stable elements 18, and biological parameters 1. Totally 42 parameters we are carried out in this project, but we are in this time only we are explaining the chemical parameters part only in this project. This is the study area. There is the Indian map, Andhra Pradesh, and the circle. What is the circle area is our project, proposal project, Povada site, Ranasthalam, Srikakulam district. There is a Srikakulam district in Andhra Pradesh, Uttar Andhra Pradesh. This is the study area. The blue color shaded line is the sea, and the project proposal area in the sea, there is a 10 sectors divided the 0 to 30 kilometer range. 0 to 30 kilometer range. Every area is divided into 0 to 1 kilometer, 3 kilometer, 5 kilometers, 15 kilometers, and 30 kilometers. So we are observing the quality of the groundwater in the sea and 0 kilometers to 30 kilometers of the surrounding area of the Povada area. Because after establishment of the nuclear power plant, what is the changes of in atmosphere, air, water, soil quality, observing every 2 years for the environmental purpose. If the radiological parameters in low level before the establishment of the plant, if any leakages of the nuclear power plant, the radium and uranium, thorium nucleates are entering into the high concentrations in the environment, water and soil. And after that, we are also analysis of the after 1 year and 2 years or 3 years, every year the quality is observing for the monitoring of the pollution and monitoring of the human health, because sometimes the plant is not in good condition. The radiological parameters are radio waste in the form of soil, air or water. It is entering into the groundwater. The groundwater is polluted by the radionuclides. The radionuclides is damaging the human body in so many affecting of the organs, important organs, kidneys and lungs, and also heart. Due to that reason, the government of India planning before establishment of the power plant, the carried out the evaluation of the water, soil and air, soil and air. So the study area is divided in 16 sectors. This is also explained in background of the power plant. The electrical energy plays a key role in industrialization in any industry. It is used for the energy and means of electricity. So that's why the electrical resources, natural resources of the electrical energy establishment, the major non-renewable essentially available natural occurring sources of generation of electricity by the coal, gas, and the petroleum products has gone into a critical position. Availability generally, the coal and the natural sources of energy farms is day by day it is decreasing order due to the population increase and industries increasing. That's why the government of India to look after the other sources of the establishment of the power plant by the industries and also and household energy by using the power plants in radiological power plant, nuclear power plant. The object of our project, the main object of the power project is characterization of the surface and subsurface hydrology and hydrochemistry of the proposed NPP site. The establish the baseline data means what is the baseline establishment of the baseline means the first evaluation data and second evaluation data, third evaluation data like this. The evaluation one evaluation to another evaluation, if the parameters of the any contamination parameters, if increases, the government take decision how to reduce and how to save environment surroundings of the nuclear power plant around the 30 kilometers area is the surrounding area. If any nuclear power plant or any power plant, this is the study is carried out before to establishment of the project. In this project, in the nuclear power plant, the 30 kilometers, the effect of the power plant and radiological waste materials influence the 30 kilometer range of the environment. That's why the government of India, DIE, proposed to 30 kilometers radiation of the proposed nuclear power plant to Kovada, Andhra Pradesh. The study of the subsurface characteristics of like geology, lithology, groundwater table. What the groundwater table, there is a underground water table is there, water like table. The what according to the water table, the flow directions, upward direction and take upward direction. If the direction is carried in the characterization of the water table, if the canals and the pipelines, the underground drainage system in the according to the water table, establishment of the drain water system. The scope of the main object of the project, radiological and chemical groundwater surface, 30 kilometer ranges, and more focus will be given to the 5 kilometers water quality parameters up to 30 kilometer radius. The power plant is established in the sea near sea. The water quality affecting by the surroundings areas more affecting or more influenced by the power plant, the distance will increases, the contamination and the effect of the water quality decreases. So that's why the 5 kilometers, 0 to 5 kilometers range is the what is the water quality is very essential, important to the radiological parameters, pH, salinity, and ORT, temperature using sensor. And all water samples will be analyzed for the uranium content using fluorimeter. There is a fluorimeter around the cost of 15 lakhs by using the fluorimeter, by observing the water in the uranium content of the groundwater or soil or any air. The fluorimeter, it observing the fluorescence techniques like UV spectrophotometer and UV spectrophotometers like the fluorescence techniques, the fluorimeter by using the concentration of the ppm, concentration of the uranium in groundwater in observing the fluorimeter. And also ions also analyzing the ion analyzer and the other parameters also by the DBO is the estimation of the standard estimation, ORT, that is the one electrode there, ORT electrode, and pH electrode, EC electrode, TDS, EC versus TDS also calculated by the according to EC. And zinc, nickel, the trace metal analysis is carried out at Andhra University. There is a ICP-MS inductively coupled plasma chromatography ICP-MS technique in Andhra University. There is a one sample. It is the instead of the one sample in the mission, it gives the around 25 trace metal concentrations ppm in at a time. That is the analysis of the carried out. So the geography of the area. This is the geography of the area, the Kovada, and wells locations. This is the dotted area. Some dots are available in this map. The dots are is indicating the wells locations, sectors, and radius. The red lines sectors of the study area, It is divided into 10 sectors. 2 sectors, half in sea, half in land, and the remaining 8 sectors fully in the land. So the climate conditions also, we are measuring the study area, the climatic conditions. So this is the rain gauge stations around the Kavada and PP style. We are also rainfall data establishment for the record purpose. So what is the rainfall status? We are the recorded by the already establishment of the rain gauge stations are there. This is the map showing the stations. The rain according to the standard stations, the stations every season, every in rainy season, we are the establishment of the rainfall data for the record. This is the drainage system around the Kavada district. So drainage system also showing the facial diagram. This is the diagrams or facial diagram. It is the drawing by the software. The diagrams are designed by the software with GPS software. There is a software. In Kakinada, the NIS National Hydrology Department is there. There is the department in experts for the facial diagrams in any area or any situation. It is like the drawing skill. Okay, this is the water table. The arrow marks showing the water flow directions. According to water flow directions, the drainage system establishment in any area, if the drainage system, the rainwater, any place or any canal, it is finally it enter into the sea, because sea is the low level, because some areas are in high areas or low areas, any area, the groundwater, according to the groundwater table, we are study the groundwater table, what is the flow of the upward flow of the downward flow of the in underground. There is underground, there is a water flow is different from the land. So we are studying the underground water table. The water table, it is help to the establishment of the drain system and drainage system around the power plant. So basic water quality parameters. Okay. The graphical representation, the tables are many tables. Every table you can explain so many time taken. So that's why I am explaining only graphical representation for easily understanding and time take process. So the parameters of the chemical parameters we are observing in the pre-monsoon and post-monsoon, because the chemical parameters in monsoon season, the groundwater table is very high, increasing the groundwater levels, because the rainwater absorbed into the ground underground. So the concentrations of the such elements diluted in the rain season. That's why the levels of the contamination levels of the basically in low level in rain season, because the water is diluted by the rainwater. Rainwater is a pure water. If the rainwater is mixing with the groundwater, the groundwater contamination elements or concentration is diluted by the pure rainwater entering into the or mixed into the groundwater. In this, the TDS, total dissolved solids, the contamination of the total dissolved solids. It explained the graphical, the red line showing the representation of the dissolved solids ranges from the pre-monsoon and the post-monsoon or monsoon seasons. The frequency distribution and cumulative frequency distribution of the total dissolved solids. The cumulative frequencies is the standard formula there. The total parameters of the around 200 samples. The 200 samples divided into the cumulative frequency, a single graph for the easily identification of the ranges or changes of the total dissolved solids. Actually, this is graph is 200 samples. So the ranges of 500, the ranges of 5 to 1000, 1000 to 1500, 1500, 2000. So the influence of the groundwater in the rainwater, the frequency distribution, cumulative frequency distribution of the total dissolved solids TDS in a pre-monsoon season around the Powada site. This is the post-monsoon season. The graphical representation, it shows the curve explain how to changes by rainwater. The TDS level groundwater sample collected around the Powada site, it is the ranges from 102 to 3,425 ppm and 102 to 3,067 ppm in the pre-monsoon and the post-monsoon season respectively. This is the figure. The DD level water samples around the study area from the 500 to 1500 ppm both in pre and post-monsoon season. The remaining pre and post-monsoon seasons, 91% and 92% of samples permissible limit. Around the 91%, 91 to 92% of the samples permissible limit. That means the waters can use it for the drinking purpose. If the previous ranges above the 1000, that waters cannot be used for the drinking purpose, but used for the other purposes and washing and domestic aquaculture purposes. But the study area only 8% of waters only, it is not suitable for the drinking purpose, and 92% of water samples is for the permissible limit, within the permissible limit ISI standards. So the frequency distribution cumulative of the electrical conductivity and TDS, it is the both are same influence. So the frequency distribution, cumulative frequency of the salinity, if the conductivity increases, the conductivity in the TDS and salinity, both the parameters, interdependent parameters of one another. So the ranges of 500 to 1000 ppm, both the pre and post monsoon, 88% and 88% of samples are within the permissible limits according to the salinity. The parameters of salinity, it is also explain the water quality. It is for the uses of applications for the what is the parameters, what is the ranges by using the water quality parameter. This is the cumulative frequency of oxidation reduction potential. If the total dissolved solid content is increases, the potentials also increases. That's why the 97% of the ORP oxidation reduction potentials are within the range in pre and post monsoon season, and remaining samples in the above the season. So this is the facial diagrams of the total dissolved solids which is the red mark indicating the danger or the high levels of the parameter. This is the GPS well related facial diagram. This is the study area. The yellow mark indicating the 500 acceptable limit of the TDS. The green mark near maximum permissible limit. The green mark indicating the maximum permissible limit. So the red mark indicating the unsuitable for drinking purpose, unsuitable for drinking purpose. So yellow water is very good for the drinking, and the green indicates it is the maximum limit, means that is 500 to 2000 maximum limit low. So that's why it is not good for drinking purpose. Only yellow color waters are very good for the drinking purpose. The red color water is not suitable for, not used for the domestic purpose and as well as the drinking purpose. It is the TDS facial diagrams explanation. This is also total alkalinity. A yellow color, this is the 200, below 200, and the green color 2 maximum permissible limit, and red color above the permissible limit, that is 600 above the permissible limit. So the yellow color is the suitable for the drinking water, and green color suitable for the other purposes, domestic and aquaculture and agriculture purposes. Water is suitable, but not good for the drinking purpose according to the facial diagrams, the values showing above the permissible limit. This is the total hardness. The red color above 800 hardness water. There is no some water is hard water and soft water. If the hard water, it is not suitable for the drinking purpose. The soft water, it is used for the drinking purpose. The ranges of the hardness 0 to 200 range, it is acceptable limit for the ISI standards. For the drinking purpose, the acceptable limit of the hardness, total hardness, is the 0 to 200, and 201 to 800 the maximum permissible limit. 0 to 200 is a very good water, and 6 above 800, that is a bad water. It is not used for the domestic and it is not used for the drinking purpose. It is only used for the agriculture and other purpose or industrial purpose. The calcium hardness, magnesium and calcium total hardness. And another parameter, statistical tools by using explain the quality, water quality parameters. There is a correlation coefficients. It explains the what is the quality of groundwater quality parameters, because the quality explains so many variations, and this is also one of the important parameter, the correlation coefficients. It explains the water quality. It is the mathematical tools for the explain in chemical science are in explain the quality parameters by using the mathematical or statistical tools. So pH moves in the lower positive correlation. If the degree of positive correlation, salinity to the low degree, negative correlation with the parameters in post-monsoon season, low degree negative correlation, So the positive correlation, negative correlation, it explains the total dissolved solids. Pre and post moon season TDS is the high degree positive correlation. High degree positive correlation means the value of the correlation is high. The negative correlation is the value of the parameter in the range or low level by using the statistical tools. Okay, summary. A part of the project study, total number of water samples in 178 samples in post-monsoon season, 200 samples from the monsoon, pre-monsoon season. We have collected an analysis for the total samples in the Kovada district area, the groundwater characterization of the water sample during the monsoon, post-monsoon season. So the BAE BRN project study related to the present Kovada anti-pesticide research project has been divided into four parts. Part one is the deals with the introduction and the water parameters and water utilization of the various parts of the measure inequality and the quality parameters. Part B deals with the experimental methodology. There is a chemical parameters. Every chemical parameter is standard experimental procedures are there. For example, temperature, it is the temperature thermometer and ORP electrode, and total hardness, it is a total hardness standard procedure in the volumetric analysis, and DO, dissolved oxygen, and volumetric analysis are there, and chloride, silver nitrate analysis standard procedure. That is experimental procedure methodology or experimental procedures. We are following a standard experimental protocol procedures because the government of India some establishment standard protocol for the following of the experimental procedure. Part C deals with the results of the water quality and the radiological water quality parameters like radium, lead, polonium, cesium, and uranium. Uranium, the following radioactivity elements also we are the analysis, the radioactive analysis in the BRNS laboratory, Bombay, because in our area, all radiological analysis are not available, only uranium and lead availability in our laboratory in remaining parameters of the radionuclides analysis in the government of India labs. So if the project sanction, they also the analysis also supported by the government of India. This is very important in any degree college level. If the NAC grade or some of the basic instruments are available in our laboratory, can apply the government of India sanction the projects. Basically, when some important instruments are available, if some instruments are available in basic parameters, we are doing the basic parameters, they can support the financial assistance for the simple projects or like major projects. Part D, it deals with the reference section. What is the reference and what is this is the summary or a conclusion in this project circumstance. In the Kovada district, it is proposed BAE BRN project study Srikakulam district. The study area characterization of the groundwater and microbial content MPN, most probable number of microbial content count, and also present pathogenic bacteria species like E. coli, Enterobacter capsula in groundwater. So there is a groundwater in a small bacteria also present or contaminated in the bacteria contamination also we are observing in simple laboratory techniques. There is no costly, there is a very cheap techniques are available in our normal laboratories. So the findings of the project, the electrical conductivity values and the pH values, some levels of the some stations, some high ranges, the groundwater collected in the pre and post monsoon season in Kovada district rural area, and total hardness, calcium, magnesium hardness levels in case of majority groundwater samples collected in the pre and post monsoon season, 16 sectors as well as below the BIS WHO limit. Very few samples are crossed the permissible limit, indicating their unsuitability for a domestic uses. Total alkalinity levels in majority samples within the permissible limit, and dissolved chloride levels, maximum number of water samples collected in the study area. The very few samples in high level because the industrial water in mixing with the certain stations. That's why the bleaching reagents are involved in the groundwater. That's why the ranges of the chloride, some of the stations, the values increases we are finding. And the fluoride levels also, fluoride, sulfate, majority of the waters are within the range. And the heavy metals, calcium, magnesium, potassium, sodium, the heavy metals are in ISI standards, lead, cadmium, nickel, cobalt, barium, cesium, mercury, arsenic, and chromium levels within the permissible limits, because the trace metals are heavy metals of the groundwater. There is no trace metal, heavy metal contamination. We are not observing our study area in 200 samples. The groundwater collected from the parts of the 16 sectors are observed in the MPN count, the microbial count in high ranges, some stations, because the drainage system, that there is no proper drainage system in rural areas. That's why the wastewater, the washroom waters or toilet waters mix the groundwater in some stations. The microbial contamination at a few stations, nitrate contamination also increases due to the sanitary system. There is no proper sanitation system. That's why the concentration of the groundwaters in high ranges in nitrate and microbial contamination we are observing in the study area. Conclusions: the pH, the ranges, the chemical parameters ranges. Basically, mainly the pH is TDS. We are observing very few locations, high ranges, majority of the locations within the permissible limit. So the permissible limit change year by year or change in season to season. So many influence factors are there. So this time the area characteristics of the environmental characteristics of the proposed Kovada site like this. After 2 years, these levels of the concentration of the contamination levels within the chain, some stations increase, some stations decrease. If the industrial influence or increases the water quality damaging or the increases of the polluted contamination in the groundwater. So this is the financial status of the sanction in the equipment to 18 lakhs in staff salaries and 4 lakhs 8,000 technical assistant charges, travel grant totally, and 42 lakhs 8,000. This is the laboratory facilities, basic laboratory techniques in the our laboratory. This is also water collection in the purpose of during the water bottles also standard water bottles. We are collecting the water samples and immediately basic parameters, safety parameters, pH, EC, TDS, and DO analysis in the station only, because if the DO values changes after time taking, so that's why the DO and EC, TDS, safety parameters, that is the immediately take the groundwater sample, immediately analyze the some parameters, that is safety parameters. So we are observing the also this is the sample locations and sample collection. This is laboratory technical assistants. We have carried out the analysis. This is the microbial analysis of the microbial technical assistants are there. This is the fluorimeter 0.2 m, the 14.5 lakhs, the worth of the fluorimeter. It is observing the uranium analyzer or uranium observing. This is uranium analyzer. This is the alpha beta counting system. This is a water sample. If dry and take a sample dry water sample, we are observing alpha, beta, gamma count for the radionuclides. This is the GPS. We are collecting the station. The GPS values also important. Anybody cross-check the water samples details in the according to the GPS coordinates. This is the digital radiation survey meter. Digital radiation. If any area or any ground, the radiation levels we are observing high ranges, there is some problem in radio pollution. That's why basically in identification of the radio, digital radio survey meter. Thank you for the opportunity for the my dear friend Karunakar.
Ramarao, what are the methods that are used for biological parameters, sir?
Biological parameters, there is a DO, there is a dissolved oxygen, there is standard parameter.
Are you find out the microorganisms?
Microorganisms, this is multiple test tube methods in microbiology laboratory, simple microbiology laboratory. In some 2 years back, you are also doing some microbial organisms, MPN count, your laboratory. Okay.
Okay.
Or pH and EC are standard methods, but ionic electrodes are there in ionic analyzer. Chloride to fluoride, nitrate to sulfate. That's why the ion electrode is only one ion analyzer. The electrodes are changes, electrodic standard solutions are changes. What do you want the electrode? You change the standard solution and get the particular ion value.
I request.
Okay, sir.
Okay.
Okay. Hello.
You proceed, sir. You proceed. Please.
Hello, sir.
Please proceed,
Sir.
sir.
Okay. Okay. Thank you, sir, for your presentation. Now I invite Dr. Padma Madam to present vote of thanks for this morning sessions. And after that, we'll meet sharp at 2:15 after lunch with same link. Over to Padma Madam.
Yes, sir. Thank you, sir. It was a really wonderful lecture which was delivered by Dr. D. Ramarao, sir, Principal, GDC, Konduru, Kakinada, Andhra Pradesh. Myself, Dr. D. Padmastam, Professor of Economics. It's my privilege on behalf of organizers and Department of Chemistry and especially Department of Botany and GDC, Sadashupet, conveying vote of thanks, sir. Actually, Ramarao sir's project, I can absolutely say it was wonderful study projects, sir, which were done by you. As in economics, as a faculty of economics, we are always tells about economic growth and development, which includes the urbanization, like what you discussed earlier, urbanization and industrialization, which needs electricity as a key role, which plays your study area, that is evaluation of ground and surface water quality status in nuclear power project in and at Srikakulam. You discuss, sir, your project majorly focused on the effect of this radiation and uranium on groundwater as well as soil and air also. So these levels which are showing very unsuitable, that is unsuitable for mankind, which impacts badly impacts on drinking water and irrigation even. Like you, sir, elevated that which is all parameters which are excluding, I mean, which is more than the desirable ones. So the government, sir, is it this project is functioning there, sir? Nuclear project.
Madam, this project is the change of government. The project proposed Manmohan Singh.
Okay.
Change of government of India, they are planning some technical objection, that is the funding.
Yes.
And some it is not establishment, it is under process.
Okay, still they are pursuing.
Pursuing, because years back corona attacking, there is some troubles.
Okay, somewhat these people are so lucky, at least these days. Which thank you, sir. Your lecture is very immense one. Thank you for. Thank you. Over to Vinay Kumar, sir.
Okay, thank you one and all. And we'll meet after lunch break sharp at 2:15 with same link. Have a great lunch. Have a good day. Thank you.
Okay, Karna.
Hello, sir. ఓకే, ఏం చేయాలా సార్? ఏం చేయాలా లింక్? మళ్ళీ ఆఫ్టర్నూన్ ఆన్ చేయొచ్చు. ఓకే.